So how is this going to work? You're gonna manually switch the chess clock for us every time we. So okay, thanks.
And we're starting with your questions. What was the decision?
I think we were started. I was gonna say some things in the rebuttal, and I'm not planning to talk forever. I just
Okay.
was gonna do the 5 minutes or less. I don't know. Um, are we okay with that? Or do we want the judges to go first? Okay. Yeah. So I want to start with Xinfadi market because we're in disagreement on that, and that's just one of the last things you were talking about. It sounded like you were saying that's like community transmission that shows up in a market. Is that correct, sir?
I'm not sure I understand the question. I mean, we're saying the market was the next major cluster since lockdown.
Yeah, but like, do you think it's in Beijing? Do you think the virus is going around Beijing among people, but then it shows up at the market because markets are a likely place for outbreaks to
Yeah,
start?
so probably a few people got infected, and when it got to the market, amplification was faster.
Okay. So I actually hadn't read that much about this until I saw your slide. So I read a bunch of papers about this weekend, and it was traced to seafood at the market. It was traced to imported seafood in one stall. And then when it got out of the market, the cases clustered on the market. And when you do statistical centering techniques, you get, this is not exactly the same kernel density estimate that they did in Warabi, but you get the same result that the outside cases cluster on the market. So we only have two data points now, it's Huanan and Xinfadi, but they show the same patterns that you get some animal product that comes into a market and it spreads outside in similar ways. And and you get clustering within the market. It's a little better in Xinfadi. You know, it's a really nice heat map on the stall that the infected seafood was traced to, and all that imported salmon tested positive. But you do have additional hotspots, so it's not 100% clear. There's some papers the judges can read about the infected salmon, and you gave some numbers about like there's more cases in Xinfadi and the ratio is higher. I looked it up. Xinfadi is the biggest market in Beijing and actually all of Asia. It's 21 times larger than the Huanan market. It's got 20 times as many visitors, assuming the 10,000 number you said it's 200,000 visitors there. Also, they shut it down really fast. I think the cases were detected June 11th. They closed the market 2 days later. They shut the schools. They canceled flights. So the ratio is going to be higher because you detected a lot more market-linked cases when you shut down the outbreak really fast. So I don't think that proves what you're saying. I think it actually is a really great similarity to the Huanan seafood market. But just in general, I wanted to talk about probabilistic arguments. And I think it's funny that you came up with it's like 1 in 100,000 against zoonosis, and I came up with it's like 60 billion in favor of zoonosis. And what we both did is we just multiplied numbers together. So there's still human bias here, which is you decide which numbers you multiply together. And there's a huge possibility for confirmation bias there, where you get attached to one theory and you only pick the numbers for that theory. I don't know how we resolve this when we have two completely different probabilistic estimates. I think what we need to do is fact checking to figure out which numbers are important. And I think your presentation showed why the root claim might be going in the wrong direction here, is you don't do fact checking, and there's a bunch of cases where you messed up here. You said that this December 8th case happened before the market outbreak. Both sides of the debate disagree with that. You got the two visitors wrong. You're focused on these early Weibo cases when there really aren't many or maybe any of those in December. You said Warby's data is confounded by population density. You have some theory for why the case search was biased, because it was before January 18th, but actually the cases were found after January 18th, so that there could be some source of bias, but it's not that. And you ignored the early case search where it should be unbiased because it hadn't been identified yet. The excess days, you said something about 90 missing early cases. I can talk about why that's not true. You've also said it's irrelevant when COVID started, and I don't think that's true. I mean, when you want to...
Sorry, Peter, can you go back to the previous slide?
This one? Yeah.
The one where you were making claims about fact checking. I just hadn't finished reading.
This one, yes, sorry.
No, yeah, that one. Thank you. Retrospective. Okay, I'm going to be interested to hear what Saar has to say about this. Thank you.
I think these are the kind of facts that we need to go over, because we can't do a probabilistic argument until we know what the right facts are. We need to know which facts to put numbers onto. We can't just start with the numbers. And I think when it started is actually really important, because you want to know where and when. Those were like the two key questions I started looking at when I was trying to figure this out. And there's a lot of little things like the December 8th case that you missed. But then 2 slides later, you're talking about Mr. Chen on December 16th. That's the same guy, but you didn't notice that you've double labeled the same case. As far as I could tell, I might have just been misspeaking. You talked about this Texas sharpshooter fallacy, and I think this is actually a great metaphor, but I think this also applies to the Mahjong room, to the toilets theory, to a lot of these things, to this Weibo data, where you describe this as unbiased, and I think there's a bias you're missing, which is that you've got to have like 20 different ways you can map the data in Wuhan, and most of them are going to say north side of the river, but you're going to find one that says south side of the river. And the people who are doing motivated reasoning are going to latch onto the one that says south side. That's where the bias comes in. When you actually look at that data, it's from February. It's people putting the dates in retrospectively. 3 entered December. That's kind of weird. That could just be old people fat fingering the date. We don't know which of these 3 points are from December. It could be the 3 points right there. I tried to find a copy of that data. I couldn't even find December points in it. I only found January points. The Weibo data in general, I totally agree with your point that this matches the population density of the city. This is Weibo, this is density, I think this is elderly density. But that's not true for the early cases. You can match these contours of the early cases, and they don't match the density. This is the clearest way I found to show it, which is you just draw the points on it. And, that probably explains the stuff here. I don't think it explains the cluster there. I think there's still a meaningful pattern there. So I guess I'll stop there. I don't want to talk the whole time. I want to understand why you think the case search is biased. What your best arguments there are.
Wait, what is biased?
Why the early case searches? You think it just looks like it's centered on the market, but it's not actually.
Right, so it's important to I need to change the clock, right? Or when he's asking this time, I don't know.
I thought Peter was still asking you as part of it, so
if he's asking us this time, okay.
That's
I don't remember the rules. Okay, so.
Well, I can just talk for 30 minutes, but I think it'd be a better discussion to here's a disagreement. Why do you think it's biased when I think it's unbiased?
Okay, so it's important to understand, when we say biased, we're not saying that the market is just a random place. Okay, we're definitely accepting that it is a.
Wait, sorry, is this counting against my time while he answers?
I don't know what are the rules.
So in the rules it says, if you ask as I understood it, Peter, if you ask a question of Saar, the time counts against you because you have the floor. You can yield the floor and have him start his rebuttal and answer that question. I would be obviously totally.
Well, so there's 2 incentives here. One is I could just give 30 rebuttals and that's going to be a crappy debate. Or I can say, here's a point, I'd like you to respond to this and yield. And I guess he could switch to a completely different point if he wants. But I don't want to lose all my time just based on he's answering questions when I'm trying to have a discussion. So I think we need to resolve that.
I'm fine with switching the time if that's okay with you, Saar.
I'm going to do one of 2 things here. I'm either just going to say a bunch of complaints or I'm going to yield. But I'm not going to give him my time while he answers his questions. Or answers my questions.
Totally understand.
I mean, if the idea is that I answer a specific question, then
or
don't
yield. Let's go with that. It'll be a good discussion. I yield my time. Here's what I'd like you to talk about, but obviously you can talk about anything.
Okay. So I will answer maybe in a different order. So first of all, the most important thing is about the billion versus 100,000 probabilities. Yes, many people do probabilistic models and get very extreme numbers. Okay. In the case of the in the case of the murder we analyzed, we had some law professor who did an analysis and showed that it's 1 in 200 million probability that the person in jail is not the murderer, right? He by the way, today he agrees he was wrong. So doing bad probabilistic calculation is very easy. What we do is we go through a very careful methodology where we steelman each hypothesis, find the best explanation, look for dependencies, look for the best numbers, give a low range and a high range, and integrate all the numbers to something that is robust. So it's important to understand, we're not saying it's a 1 to 100,000 probability. The result we have was, I don't remember, it's 99%, that's 1 in 100. The 1 in 100,000 is how much you need to shift the data in order to pass 50%. Because remember, we do have all these safety measures, right? So as you, it's not enough, if 99% doesn't mean 1 in 100, and if you give 1 in 100, it's, you win. You need to go over all the safety measures, and that's 1 in 100,000. Okay, so that's in general about the probabilities. Yeah, we're perfectly fine in getting some facts wrong. We corrected some numbers based on your analysis, and it's perfectly fine that we will correct more numbers and eventually concede that zoonosis is most likely. We don't have a problem with this method. Just point out any mistakes you see, and we will correct. At this point, the amount of evidence is so strong, I can't see that happening, but sure, let's do that. About specific things you said about Chen, if you go back to the recording, I said specifically that some say 8, some say 16. This is not settled. 16 is not settled. It's based on an interview that is not clear who they're talking about, translated. We're fine saying it's 16. It's not an issue. But notice that when I said 16, I mentioned that some say it's 8. I did not neglect that.
Sorry, I must have missed that.
Okay. About Mahjong being a sharpshooter, no, Mahjong starts from lots of testimonies of people saying Mahjong. It starts from the evidence. Didn't start from looking for what could match. Okay, it started from the evidence of many people saying the first cases were Mahjong players. I agree about what you said about the south side and the north side or east or west side in Weibo. The data we brought about WIV being in the center, we explicitly said this is not something we believe in. This is just showing you how easy it is to cherry pick data. We're agreeing with you. That's sharpshooter fallacy to claim that Weibo supports WIV. The idea was to show how much you can play with the data and how unreliable these things are. The thing about Weibo being later cases, sure. However, it's important to remember that the whole idea of the market analysis is that cases do spread geographically, right? So either we accept that cases spread geographically and gradually, or we don't. So if the Weibo data centers on the population center, then that's evidence against it being the market. So we need to decide whether the centering works or not. And it's very important to understand about the bias. We are not claiming the market is a random place. We're saying it's a significant superspread location due to its features. Okay, and it's definitely a place where a significant number of cases were. Probably not 33% as claimed, because there is the biases of market being noticed by people, by health worker, because it's been a criteria, because contact tracing, everything. There are a lot of things that will age, a lot of things that make the market more likely to be noticed. But we're not saying it's not important. We're saying it's one of the main superspread locations, and we're even okay saying this is the first big cluster. We're fine with that. Just say that there were many other cases spread around the city that didn't mature into super spreader event. That's all. I yield.
I mean, how many other cases would you estimate there were? So let's say December 13th, you've got these 5 hospitalized cases at the market that I pointed out. 4 workers, one's a repeat visitor. And so you have more cases than that probably because those are hospitalized. But how many cases do you think are in Wuhan on that date? A yield?
I don't have a specific estimate, but probably mid-December. I don't know. It's probably 10-20% of the cases are in the market. Maybe less. I don't. I need to think if you need an exact.
I do need an exact number because that clarifies how biased it is.
What exactly do you need?
So this is
estimate?
December 13th. So there's these 5
December
hospitalized
13th.
cases at the market, and you think there's lots of COVID elsewhere in Wuhan.
So
How many
there's
total?
five hospitalizations.
Yeah, let's say
And you
hospitalization
want to know how many cases overall in the world at that time?
in Wuhan.
Yeah, it's the same thing. Yeah. Okay, and we'll try to get that. Okay.
No, can you answer that on the spot?
No,
Even order
research. I can
even
do
order
this.
of magnitude. Do you? I mean, do you?
Order
so,
of magnitude, I would say 5 to 10 times more.
So 5 to
50.
10 times 5, so it's 25 to 50 cases, or 25 to 50 hospitalizations.
So remember that the market is older population. So if you want it in cases, yeah, probably 50 to 100. I don't know.
Right.
But
So
it's not something I can do.
if
I need to think
there's
about
100 cases and the hospitalization rate of COVID is 5%, then you've already proved it's the market. Because
this
why
is
would
an
the
addition. This is an addition to the market. So
Why would
I
the
would say,
5%?
if you want, I'd say 5 hospitalizations in addition to the 5 in the market. But really, you're trying to get something that I'm not claiming to have the entire model of the outbreak in my head. What we do is we look carefully in data and do probabilistic analysis and collect information and correct when there are mistakes. There's no use trying to push me to give numbers.
you're thinking it's double, is that what you said? There's 5 hospitalizations in the market and 5 outside the market?
It's just a guess. If I can,
Or do you think
I'll
it's like
give you
10
the next session? I can give you numbers in session 3. I can't give you something off my head now. It's not how we work. It's not about
Okay.
It's not about us being smart. It's about the methodology.
Let me just explain why this matters. So here's a model. We know how fast COVID doubles, the epidemic doubles. It's about 3.5 days. So that's 2 doublings per week. So let's say, let's guess it's 50 cases at the market, or 50 cases total on that date. Hospitalizations is 2 to 3. And then you can apply these doublings forwards and backwards. So you can figure out when it started, November 15th roughly, November 22nd maybe. So that lines up with PCAR's model pretty well. And you can apply it forwards to when the Wuhan lockdown started. And these numbers, they're correct to about a factor of 2. Now, you can also do this with, and so here's cases and the lockdown, and you can cut this, it's like an exponential curve until it hits 123. And then you can fit an exponential curve to that data, and it's, r squared 0.999. It's a great model. And then it slows down when the lockdown happens. Now try 1,000 cases on that day. You project it forwards, and you get 10 times as many deaths as actually happened in Wuhan. So it can't be 10 times as many. The market needs to be like all the cases or a substantial fraction of the cases. And if you went even more crazy, like 10,000 cases on December 13th, you're going to end up with 400,000 deaths in Wuhan by the time the lockdowns. And this is bigger than any death total of anywhere in the world. And even this one, I think, once all lockdowns are done, it's going to be worse death total than any city that hit the world. And you say that got hit by COVID. So this is actually a case where human reasoning fails. People don't understand exponential math intuitively. They think, September origin, November origin, doesn't really matter. It's just going to sputter along. But every time COVID gets somewhere, it either goes extinct right away or blows up exponentially. And a difference of a month is a factor of 256 in cases. 2 months is 65,000. And if your model can't distinguish between like October and November, you're ignoring a huge fraction of the data. Your model doesn't explain that very well. And so we can bound how biased either, if we go back to the one that works, where let's say it's 50 cases, 2 to 3, maybe it's really 100 cases and 5 are at the market, something like that. And maybe it's more hospitalizations because they're older, that would work. Then it's like every case is at the market. So you can still have a lab origin, because you've had 50 cases by then, and case number 1 could have come from the lab and he just went straight to the market. That's possible, but is highly improbable. And you can't have the scenario where it's just all over Wuhan because it doesn't fit with the math. And I mean, I'm not just making up these doubling numbers. I've looked these up over multiple sources. I've checked them by fitting the data in Excel myself. Yeah, I yield.
I'm going to interrupt before Saar has a chance to speak. One of your slides, I think you were showing real data. Can you clarify if that was cases or detected cases? And if so, like how big of a disparity? This slide.
This is detected cases, which are mostly hospitalizations, honestly, at that point.
Okay. Do you have any notion of roughly how big a disparity is between detected cases and actual cases?
It's roughly it's on order of magnitude of the hospitalization rate.
Okay, so you think the real numbers are 20 times what's in this plot?
Yeah, something like that.
Okay, thank you. That's all I wanted to
20,
ask.
yeah.
And let's hear what Saar has to say.
Sure. So again, it's important to understand our position. We're not saying that the market is not important. We're not saying that the infection there is like the general population in Wuhan. We're not saying it's all over Wuhan, lots of infection. That's not what we're saying. We're saying that the evidence shows that even though the market was an early super spreading event, it was not due to an animal. Okay, the whole analysis I walked through in the end of my my part was about showing that the evidence doesn't match the hypothesis in a way that would support an animal being the one bringing it to the market. So it's important to attack what actually we're saying, not something that other people are saying. So we are definitely willing to accept that the market is a major event. Now, a lot of the data you showed is data I'm seeing for the first time, so it's hard for me to respond. So I'll look at it.
Because you didn't fact check. Like these are basic things you should know from the exponential math in Wuhan. You should know the case numbers and the death numbers if you're trying to understand how it started. Sorry, I shouldn't be so aggressive. Maybe let's talk about the Mahjong room a little bit.
This is my time now.
Oh, sorry. Okay.
Okay. Yes. So So it's important to understand what we're claiming about the market. So a lot of the data I haven't seen. It's new to me. We need to analyze it. It takes time. And maybe we'll find it convincing. I'm not saying anything, but at this point, I can't respond to a lot of it. First instinct about what you said about the exponentials being unintuitive. So I think what's missing there, I need to verify. I think what's missing there is you're forgetting what is the importance of a closed location with ideal conditions. By the way, your screen is still shared. I don't know if you care. So when you have a closed area with the Mahjong room, with the poor ventilation, with the humidity, with the older population, when you have that closed area with repeat visitors coming to the same place every day, you will have a much higher exponent within that than in the rest of the city. Okay, so saying that if there are more cases outside the market, that would mean there would be 10 million dead or something like that is not a correct interpretation of exponentials. You need to take into account these are different exponentials inside the market and outside the market. I yield.
Just to be clear, I did present an exponential model already. This is Picard's model from number of cases and hospitalizations. This just doesn't go past much past January 1st. But so
I'd like to try to I think both ask a question and resolve the kind of contention over this point. So if I'm understanding correctly, and this I will ask both of you to respond to in this context, but it seems like both of you agree that the market is an early super spreader event or an early cluster event, whatever you want to call it. There is, if I'm understanding your position correctly, Saar, you're thinking that, or presenting evidence that that's due to some event. Could be Mahjong, could be an individual person going to one of the Mahjong halls, it could be something else. But regardless of how it happens, you agree that that's probable, or you agree that that's the cluster event. You think it's much more probable that that's due to ultimately not an animal, but agreed there. Peter, you're claiming that it's the kind of difference in probabilities there, or the probability that you would see it emerge at the market if it wasn't via an animal, is pretty low. Or if there weren't, I think an additional part of your analysis is if there weren't a significant number of other circulating cases, which is why you're trying to press Saar on the question of where those other cases are. Is that a fair understanding of your position?
I'm saying that the simple exponential model explains that there can't be a lot of cases outside the market. And I'm not saying it rules out a lab leak, because if you do have 50 cases, patient zero could be at the market. But this is why I have the factor of 10,000 in my analysis, because it really just needs to go straight from the lab to the market without really spreading anywhere else. And that's such an unlikely coincidence. And you can come up with some other reason why it went to the market. It's not the animals, it's the Mahjong room or whatever, and we can and should discuss those things. But I think you really bounded in which lab leak theories you can make. And you can really rule out the, like, it's everywhere and it's only because they looked at the market theory.
Totally. It seems like it feels like that is not deeply in contention here. Saar's numbers, at least what he has said, seem within the realm of what I would presume the error is in many of these estimates, where we're asking about one or two cases. But it seems like you guys don't disagree a significant amount about those early cases. I understand the importance of them. I'm not making light of that. But it seems like, maybe let me pose this as a question. Your exponential model, obviously that's very, very, I guess, sensitive to small changes in numbers. Do you think that the accuracy of the data that you have for the number of cases, when and where they are, allow you to make that there's not a very large error bar in the timing? Like, basically, do you think Picard et al. Is completely accurate, I guess?
Picard used a more sophisticated model to show the same thing. And I thought about this a lot, and that's why I came up with the 2 doublings a week, because it fits the data almost perfectly in terms of the actual model. And it's the kind of thing you can just wrap your head around. Whereas if I just show this graph of, here's what this scientist came up with, you can say that scientist is motivated, he wants it to not be the lab. That's math, that's code, who can understand that? But anyone can multiply by 4. So, and you can use that to come up with pretty much the same answer. That's why I went with that, and I think it gets the point across best.
Okay, I've asked a bunch, Eric.
And there's uncertainty in Picard's model too, I should say. In all these, because it doesn't go exponential right away, there is this possibility of stuttering transmission between it not going extinct and it going exponential. But that's it can't do that for long. So that's really where you get that, and the doubling time is, there's a tiny bit of uncertainty on that. So you mix all that, and you're not going to get down to the exact week, but you can definitely get down to the month. It's not September, and you can't be off by a factor of 10 in number of cases and stuff like that. Maybe you're off by a factor of 2.
Do you want me to answer as well, or?
Totally,
So just to say a word, so I basically agree with your description. To be more accurate, we think there were a few more, maybe 1, 2, 3 more super spreader locations at the same time, but were not noticed due to that. However, our model, the market analysis that we did, does not require that. So I'm willing to just say the market was the first super spreader location, and it doesn't changes the numbers at all in our sense. So I'm fine just conceding that point. The market is the only location, but it's very important to understand about why this happens. So the market has certain conditions that make it much more likely to be an early super spreading location. And that is because of the enclosed environment, ventilation, humidity, repeat population, elder population, all of that makes the exponent within that environment much higher than the rest of the city. And as we all know, the base of the exponent is a big change when you expand it over a few weeks.
Saar, can I follow up on that last comment you're making? So to reconcile both of what both of you are saying into a single consistent story, I think what it looks like is, imagine there are 100 locations in Wuhan, one of which is this market, and the locations have different conditions, and the market has very conducive conditions for contagion and the others do not. The virus started somewhere outside of the market in the lab leak, and people at all or many of these locations were exposed incidentally to some amount of virus, and it only became a super spreading event.
Yeah, you cut off. Can you go back? Can you go back 10 seconds? Sorry.
Yes, sorry. So out of 100 locations, one amenable to transmission and 99 not. Many of these locations had people exposed, or maybe someone infected showed up one day or something like that. But at only one of them did this one infected person become a cluster of cases. It sounds like for the transmissibility in the market to be relevant, you have to have someone sick show up at the market, is what I'm saying.
Yes, of course, yes.
So are you imagining that the virus started from a lab leak and a small number of sick people between them visited many locations and one of them is this market?
Yeah, that's what I'm saying. So the lab worker has infected maybe a few close friends. They went to several locations across the town. They all infected more people. But the people in the market or in the Mahjong room, they amplified much quicker than all the rest. Okay, so after 3 weeks, they grew 100x and all the rest grew 10x. Okay, that's the point.
Yeah, that was all I had for that question. I have many other questions. I don't know if you want to move on to questions or if the two of you want to keep talking about these points.
I'd like to ask a few more, if that's okay.
You want to keep talking, please go ahead. Thank you.
Yeah. So I wanted to ask, sorry, he's focusing on this Mahjong room. Did you look up how many Mahjong rooms there are in Wuhan?
We tried. We couldn't find good data, but in general, Mahjong rooms are very popular all across China. It's a very popular pastime.
I found a paper on this, tried to get all the numbers. They found 838 chess and card rooms in the study area. And if you include other leisure entertainment, bars, theaters, cyber cafes, karaoke, they found almost 3,000 places. So what is special about this Mahjong room when there's 3,000 of these places? And I can play a video of this also. The Mahjong room is like a little closet in the market. It's tiny, it's not frequented by visitors. Do you want me to play a video?
Sure. Yeah.
This is Francisco's Twitter. That's Peter Emberek. I think this is the Mahjong room. I think this is this staircase that goes up to this little tiny closet where people hang out. Not 100% sure, but that's the closest I could find to a video. So, the traffic to that is visitors only. Outsiders aren't going to go to find that closet. And if you look at all these other leisure places, it's going to be, hugely more traffic to those. It's going to be even lower than the 1 in 10,000 that I gave you. Why is this one important? The important part is that it's for wet markets, and this is the one with the most wildlife shops, a yield.
All right. So first of all, again, this is new information. I need to look at it. In general, I think I need to look back at the data, but I think there were definitely. It was not small. There were several people there, visitors. It was not 5 people. This was dozens of people. But the point is, this was the combination of having a Mahjong room plus a location where there's other reasons for amplification. Now, it's important to understand, if we didn't have any other evidence about the market, I would say this is an interesting coincidence. Yeah, this is like a 1 in 20 coincidence. That's a big base factor. But the problem is, you have all the evidence that doesn't support the market. You don't have the animals, you don't have the animal vendors, you don't have the distribution, you don't have the RNA reads, nothing supports that. So the model I did at the end looks both at the coincidence and both at the evidence. Okay, together they push the decision to be that an animal is not a reason. If we didn't have the evidence, if the evidence was neutral and not going against the market, then definitely that was a significant factor that would maybe move it 20x. That would be a significant coincidence, yes.
Do you yield?
You were mute.
Did you yield?
Yeah, sorry, yeah.
Okay. So, did you know that they tested the Mahjong table in the market, and it tested negative?
Yeah, so I heard about that. I've never seen anything reliable to confirm that, but would love to see. If
That's
you
in
have
the
it.
2023 paper. I put that in the links in my last presentation.
Okay, so again, I need to check. I remember asking people about that. They said that there was maybe something, but it's not reliable. If you have a source, I can now look at it.
Yeah, this is copied from the paper. I pulled out that source. So there are no positive samples from the Mahjong room, but you keep describing it as like the hotspot in the market, but there's no positive samples there. And so this is a little late. This is tested on February 20th. So RNA degrades over time. You could say maybe it's degraded too much to test positive, but on February 15th, the drains are still testing positive for shop 629, and they're still testing positive for the other wildlife shop, and they're not testing positive anywhere else in the market at that time.
Yeah, I need to look at it. I mean, the idea is we're trying to do the debate in a way that the evidence is known beforehand, so everyone can prepare. You know, I can't.
I mean, it's public information.
What?
It's public information. But I'll share all of it.
Yeah, I know it's public, but the idea was to try to share the information beforehand. I can look at it. I can only look at it and
I
respond
don't know
after.
everything that's going to come up, and I only have 90 minutes, so I can't put 3
That's
hours.
fine. I'm not blaming. Just saying that you
Okay.
can't expect to get a response for something that
Yeah,
I
sorry.
don't know.
So
I'm not an expert in that field. All I do is integrate the information into a probabilistic model.
I found one other picture of the so this is outside of the Mahjong room. That it's that window right there while they're disinfecting the market. And I actually found something interesting. There's a sign on the wall, and there's a character that got removed from the sign. So I translated that. It's this Ding Sheng blank Wei wholesale store, and the missing character is probably Ye. It's Ye Wei. It's a wild taste. It's a term for eating wild animals. And they took down this character elsewhere in the market because they didn't want to admit that they were selling wild animals. So the Mahjong room literally has the answer written on the outside of it. And when they go in the WHO report, we have another picture of it, and they've taken the sign down entirely at that point. I mean, I'm not 100% saying that proves anything, but I found that interesting. I think the logic is more absurd for this like
And
toilet
information.
theory. There's millions of bathrooms in Wuhan. Why is it the bathroom in the market that matters? It's the wild animals in the market. That's the unique thing.
I'd like to focus, unless Saar, you want to respond to that. I'd like to
I'll
ask
just
a
say
question.
quickly, I agree that it's an interesting coincidence and it would be significant unless all the evidence wouldn't support it.
But this is Texas sharpshooter fallacy, because this is a heat map. This is actually just over a smooth heat map. There's not actually positive samples there, but they found that if you make this heat map, hey, you can plug the Mahjong room or the toilets right there and then say, hey, this is what caused the pandemic. But that's seeing a pattern because you want to see the pattern, because you want it to be a lab leak. You don't want it to be the animals. It's got to be something else. Therefore, it must be the toilets. But there's toilets everywhere.
Yeah, so far we're not claiming the toilets as the reason. We're claiming the Mahjong hall as a better reason.
Okay.
Yeah, that's and again, if it would have been an interesting coincidence if the evidence was not against the market.
Okay, I'd like to focus this question. Eric, do you have some, I don't want to cut you off because I've been talking a bunch, so you first pass, but otherwise I have a question about this.
If you have a question about this, go ahead. I got lots of questions, but not about this.
Okay, so then the thing I want to, this is mainly, regarding the evidence. One of the things I've been struck by is how limited the environmental and animal sample sequencing data is, and how even in those places where it does come up positive, it's either very low, there's in some of those samples, there's a couple of reads, or they've got a CT value that's like 35 in the qPCR. They've got things like that. And in general, you've got the 5 hedgehogs that have it in one freezer, and you've got different animals. This is just, I think, an intuitive reason why there is skepticism about the animal emergence and why it just like why have we not found more things environmentally or generally in animals? Can you speak to that broadly, generally?
Yeah, I mean, so there's a lot of goalpost shifting here to begin with, in as much as the Chinese said there were no live animals at the market during the WHO report, so they were, lying about that. And then the lab leak supporters, they spent all of 2021 saying, show us there's a raccoon dog. We don't even know they were there because we have Eddie Holmes' photo from 2014, and what does that prove? That's 5 years ago. And then as soon as we suddenly got this data, which China had been sitting on for 3 years, and it showed that they're not only in the market, but they're right there in that one shop, it was, oh, well, that's not important because they're not positive, or we can't prove 100%, there's no correlation, or I kind of skimmed through this too fast in my presentation, but there were a bunch of different reasons they didn't like it. Alina Chan said the human DNA had been removed from the samples, only leaving animal DNA, and that wasn't the case. And then they claimed that the sample was not positive, but it was NGS positive, but not PCR positive. So that was sort of splitting hairs because it's low amount of data. Low amount of reads, which you're correct about. So, and Saar already mentioned this in his presentation. He said, even if a raccoon dog was infected, that doesn't prove that the market's the source. So you're going to get more goalpost shifting if somebody comes up with a really clever way to analyze this data to prove that it was somehow one of those infected animals. You can still shift the goalpost on the lab leak theory to say, oh, but a person came into the lab in November and infected the market.
I understand that, but you're not debating all lab leaks. You're debating what's presented here. I think the thing that I'm trying to focus this on is I don't really care what Alina Chan says. I'm trying to understand why they're, given that an animal under the zoonotic theory produced this infection, I would imagine there has to be a high prevalence of virus in at least some animal. And I can understand that the Chinese have culled the animals, they've done things, but every sample I've seen does not have a lot of virus.
Yeah,
Why is that?
That is a good question. So this is Jesse Bloom's analysis, really. I prepared some slides for that because I thought we'd maybe talk about that, and Saar talked about it. And there's a number of issues here. They sampled on multiple dates. They went through on January 1st, and they looked for the stalls with human cases. And then they came back January 12th, and they looked at the wildlife stalls. So those are later. So you're going to find more RNA in this is the two dates and why they took those samples. And you can graph the reads over time and color code these. And the ones with a lot of reads were the ones taken on January 1st. And there's less reads on January 12th. Here's another way to graph it actually by date. So read count on the first date is high, read count on the second date is low, and it keeps going lower. So that's one problem is it's not an apples to apples comparison if you look at January 1st and 12th on the same plot. And Jesse's correlations do that. Like when he does this, when it's all dates and it says, it's not wildlife, that's comparing both of them. This one is only January 12th. And you do actually find a few animals that come out the top of the list, like the porcupines and the hedgehogs have a good correlation if you only look at the comparable samples. And yeah, you can break them up this way. Like if you only look at the January 1st ones, human comes out with the highest correlation. If you only look at the January 12th ones, animals come out with the highest correlation. And if you combine them, you get kind of a meaningless result where it's like fish, some weird stuff. So should we expect to see a correlation? For one thing, the animals, if they brought it to the market, they were infected earlier, so there's more time for it to degrade, and the people who got infected on January 30th are the one that's going to have the highest read. There's also some weird ways here where they only sequence certain samples. So they sequenced the negative samples in the wildlife stalls, but they only sequenced, and they sequenced all positives, but included also the negatives to the wildlife stalls. So that throws off your correlation. The wildlife stalls are also diverse. They're selling a ton of different animals. So you're going to get like a bamboo rat and a raccoon dog there. So if you do look at only this correlation, you do get the porcupine and the hedgehogs are the best correlation of anything if you're comparing only the animal sampling day. Is it a porcupine based on that math? I don't know. I don't think you can actually conclude which animal it is from the. If I could tell you which animal it was, I'd be publishing this in Nature, and not having this debate. This is a really important question in the world. Bloom said that there were like only one relevant sample, but he left out 4 COVID positive samples which had hedgehog DNA from that table. He also did this 20% criterion, and if you don't do that, it's hard to hit the 20% threshold because every shop is selling 5 animals, and he also included the chordates, which are the fish, not just the mammals. So it's really hard to hit the 20% threshold on that.
Totally. Yeah. I mean, I'm with you 100%. I'm hearing what you're saying, but like if you go back to that table there,
Yeah.
like they sequenced this to a depth of 1 × 10⁸. I looked at that,
Yeah.
and they're getting 124 reads. I think that's where the kind of I think there's just there's something unusual about that low a number. I'm not saying that it proves anything either way. I was just hoping that there was something that you had seen in that, because that's just a while ago.
Yeah. I mean, really the market argument is there's this really great evidence linking it to the market, this 1 in 10,000 coincidence. And this market is just like zoonotic spillover Disneyland. And that's why you would expect a zoonosis there. I don't think the data can actually tell you which animal it is. And also, we know that they're COVID susceptible. Raccoon dogs can get and transmit COVID and SARS. So the animals are there, and the shop they were in tested positive 5 times. And that's our best lead relative to, it's the toilets or it's the Mahjong room. Yeah, it's really low reads in that one sample. Jesse said, hey, how's that differ to the other one that said 0? And the one that he actually I figured out which one he picked was 0. There are 0 other positive samples in that one shop. So I don't know why he picked that. Like, if one of the ones has 1 read, but it's also in a shop that has 5 reads and the drains test positive and the drains test positive again, that means more to me. Like, if it was just 1 read and nothing else had happened in that shop, I would look at it differently. The correlation, like, people can move around the market, but the caged animals can't. So the human virus should always be a stronger signal that's gone everywhere, but the raccoon dogs are stuck in cages. So if one shop is infected, does like that raccoon dog necessarily infect every raccoon dog in the market? Because that's mostly what Jesse proved, is that if it is raccoon dogs, it's only testing positive in one raccoon dog shop, though there may be raccoon dogs in other shops that are negative. And I don't know what your model is, whether they should all be infected or not.
That makes sense. I just want to pause there. That gives me plenty of what I was asking for. Eric, why don't you go ahead?
Okay. I think I'll start with a question for Saar. For all of my questions, I'm interested in what both of you have to say, but I'm going to target at one person, and if the other person wants to rebut, and you want to spend your time on that, go ahead. I'm going to start with my second heaviest question, which is, in your Bayesian analysis, you give an update of 12 times in the impartial column for the Wuhan location, if I understand correctly. And I want to understand correctly that you're not double counting. You're not getting credit for the 12 times in 2 different places in your analysis. So in a very simplistic manner, imagine that there are 12 cities, and Wuhan is the only one doing gain-of-function research. So if it shows up in Wuhan, we get a 12 times factor. But what I want to know, and I don't know, is if in these other 11 cities, there might be other processes that are not gain-of-function going on, either in labs, maybe something nothing to do with labs, and that if the pandemic had instead started in Chengdu, then we could be having this exact same debate today, but instead of it being the lab-leak gain-of-function theory, it's something else that's specific to Chengdu. So if every city has something specific to it that you can make a tailored pandemic origin, then you get the 12x in 2 different places. You get it once where you explicitly listed it, and you get a second time when we narrowed from 12 different theories to focus on lab leak gain of function, which is implicit by the existence of this debate. I don't know if there are these other theories, so I'm interested to hear what you have to say about that.
Yeah, sure. So first of all, it's important to realize that I don't think anyone is contesting that WIV is especially active, especially risky. That's they do have the BSL-4. This is a place that is doing a much larger scale of virology research and especially gain of function. Having said that, there's no double counting here, because at other points we do talk about the specifics of WIV work. Okay, we go specifically for the diffuse proposal, we go specifically for the interests of the researchers there. So if we had the exact same virus in a different city and it would match the WIV activity, that would be evidence against. So once you focus on WIV, okay, and you look at the specific activity there, then that's it. Then Wuhan is not double counted. Is that clear, or?
That makes sense. I understand your argument. Okay, I'm assuming Peter has no comment. I have a question for Peter.
I think we'll talk about double counting more in session 3. I think there's many ways where he might be doing it, but I don't. Today I just want to figure out if it's the market and some stuff like that.
Okay.
Like, I think his priors are generally too high for lab leaks. We're off by a factor of 100 roughly in terms of how, and I think we need to establish lots of things like that. But again, future discussion.
Okay, I have a question for Peter. If I understand correctly, on roughly slide 170, 171, you're talking about having 2 different spillovers from animal population to human population. And that seems unlikely to me. And to explain why, we imagine that in this environment there are a very large number of different viruses present in the animals, in the wildlife, either specifically in the seafood market or generically across the globe. It seems like an overwhelmingly massive coincidence for the same virus to transfer from animals to human at the same time twice. It seemed, why couldn't a different virus have spilled over the next day or something like that?
Oh, that's an easy question. I mean, because that virus is the one that infects humans. So like we already established, I pointed this out in my presentation, there was a bamboo rat coronavirus in the same shop that had a foreign cleavage site, and it's from southern China. So that proves that viruses can get across China, and foreign cleavage sites are relatively normal. Though it depends, whether that's sarbecovirus or whatever. But the bamboo rat virus didn't spill over because it doesn't have the right ACE2 binding, or it doesn't have the right stuff to get into humans. But once you do find a virus that has that right stuff, it's exceedingly likely that it's going to infect more than one person. Does that make sense? Or I can give more metaphor. And this is what we expect with, like, MERS had multiple spillovers to humans. It's completely normal. And we can see this actually when COVID spilled over into mink, it spilled back into humans. And the humans are the red in these trees and the mink are blue. So once it gets on a mink farm, it does repeat spillovers back into the people working on the mink farm. So we know that's normal.
I think the mink farm is different because once it's been spreading among humans, it's had time to adapt. I imagine that after a species jump, there has to be a period of quite intense and strong adaptation as it has to adjust to the new host. And after it's adapted favorably to humans and then it goes back to minks, I can imagine in that circumstance it's much easier to get back into humans again.
Okay, so that's the question of, is it pre-adapted? And that's really a debate 2 question, whether it had this accelerated evolution when it spilled over, or whether it was pre-adapted to people. And that's an interesting argument that could be in favor of the lab, as Saar points out. Is it pre-adapted because they worked on that, or is it just pre-adapted by chance? So what I'm positing is that it has sufficient pre-adaptation to infect people for whatever random reasons, because it's this 1 in a million virus or whatever, but there's millions of viruses in nature, so there's bound to be one of them. And once you do get that virus, it's going to spill over multiple times. The actual spillover event isn't something magical. It's just, it could already do that. but once it's in the shop, it can do that multiple times. I mean, I don't know if I love this metaphor, but I think this is Joel Wertheim also on the Picard paper, who talks about it being like, Mount Everest is hard to climb, and we went thousands of years, our whole species, without ever managing to climb it. But when we finally did climb it, we climbed it twice in a day. So you could say, what are the odds that would happen twice? Well, you have all the conditions in place. You finally figured out how to do it. So 2 versus 1 is not a big deal.
Eric, I think you're muted. Oh, no, you're not.
Saar, did you want to respond to Peter directly, or I have a related question for you? I can go straight to that.
No, I agree that you should expect multiple spillovers once there's a virus infecting wildlife population.
Okay. So, Saar, on your slide 20, we're talking about SARS-1 at this point. First, something really quick. You mentioned that there were 4 lab leaks, but on slide 20, I only see 3 lab leaks.
Yeah, one of them infected two lab workers 2 weeks apart.
Do you know which lab that is?
I think that's the Beijing one.
The one that's marked China 2004?
Yeah, I think it's that one. And by the way, that's an important reference point because the whole lineage A, lineage B is is making a lot of it carries a lot of weight in the zoonosis claims. We do not think these are 2 separate lineages, but even if there are 2 separate lineages, we do have a case where that happened. We have 2 infections 2 weeks apart, which is that's 1 or 2 mutations. So that in itself is not strong evidence of against lab leak, but we'll discuss that session too. Yeah, that's it.
Okay. And then relevant to that,
Can I just point out, we are in disagreement on the number of SARS spillovers. It would take me time to get an exact number, but I think it's more than 3. Like, of these first 7 SARS cases, none of these are epidemiologically linked between these different cities. And even in the last SARS outbreak that happened in 2004, I think it's 4 cases, but they're not connected to each other. I'd have to go back over this paper to get an exact number, but I think it's more than 3.
Okay, and then related to that, I'm looking at still slide 20 where you have the 3 spillover events you show. If I understand correctly, so chronologically, of the 6 events on this slide, the very first one is the November 2002 event where a farmer presumably got sick with SARS-1 from an animal, if I'm looking at this correctly. And then it transmitted from the farmer to the labs where the lab leaks happened via research on SARS-1, and then these 2 other spillover events that happened chronologically later. Are we saying that the viruses that spilled over there are descended from the November 2002 spillover virus, or are they just independent? They've been circulating among animal populations entirely. I'm trying to understand.
So you have the civets constantly working on this virus, infecting each other, mutating. That's happening. Every now and then, a civet infects a human. Sometimes nothing happens. Sometimes secondary infections continue, and then you get an event. In parallel, scientists are constantly collecting samples usually from people, but also from the civets, working at them in the lab, and then getting infected and infecting other people sometimes.
Okay, so you're saying that the 3 spillover events you've listed on the slide are not descended from each other?
I don't think so.
Guessing.
No,
obviously we don't know.
They shouldn't be, because all of these were controlled. All of these were eventually found and isolated. So nothing descends from these 3.
Okay.
Can I just...
Yeah, please go ahead.
Do you mind if I just clarify on that? My understanding of these events were these were all laboratory stocks that were passaged. They popped them out of their liquid nitrogen freezer and they did something with them in these 3. Is that incorrect? Is there some more transmission? I thought these were all lab stocks that, they were passaging or doing experiments with.
I didn't look into the exact experiments that caused the infection.
But it's not... The critical thing is that I'm trying to understand and just making sure I didn't misunderstand is it's not there was when they traced these transmission strains or sorry, when they traced these lab-acquired infection events, they all start in the lab, or there's some potential that they started where an environmental sample came to the lab, circulated, and then went back out?
Environmental sample, I don't think so. I think these are all results of experiments.
I'm not disputing that there are SARS lab leaks. I don't think they're from outside.
Sorry?
I'm not disputing you that these are SARS lab leaks.
I'm sorry, I can't hear you. You're not what?
I'm not disagreeing that these are SARS lab
Okay,
leaks.
okay. Thank
I'm
you.
saying you can have that point. They didn't come from civets outside the lab. If that's what you're asking, Will.
Can I ask Eric one more question while we have SAR's slides up here? Okay, I apologize for this being pedantic and possibly elementary, but I think that I need to understand this well, and I think it will help the audience as well understand it. I'd like to actually share my screen and just go through your kind of initial calculation, SAR. So let me... Wow, that's so annoying. Okay, well, let's see if this works. Can you see this, guys?
Yep.
Okay, so I just want to make sure that my understanding of this is correct, and I apologize if this is stupid or incorrect. So these are screenshots from the slide SAR on the calculations, and I've just reproduced it in the Excel sheet. So we've got these cells are you trying to estimate the probability of a lab leak. Is the lab leak as a proximal cause, given that we've seen a pandemic, using data sources that are different types of outbreaks and things like that. Is that correct?
Yes.
Okay, that's correct. And then these are your final estimates of the probability of a lab leak updated for this adjustment of growth. And then you get the rounded, you just take these and round them down to produce that. Is that correct?
No.
Okay.
The two line row 12 and 13. These are the conditional probabilities of Wuhan being the location, given zoonosis and given lab leak. Okay, so it's P Wuhan given LL and P1 given Z. This is P1 given LL, lab leak, and above it's P Wuhan given Z, and it's not 10, 20, 30. It's 10%, 20%, 30%.
Right. Yeah, that was just me. I was quickly preparing this. So, okay.
Right. So now you take the probabilities you had before, you apply the conditional probabilities, you normalize, and you get the new numbers.
Totally. Okay. Thank you. That is all I needed. Okay. Thank you.
Okay, shall I give another question for Peter? I'm looking at slide 27 of yours. So here we're talking about rural versus urban. You say, based on this paper, which I took a quick look at, that the virus is 30 times more likely to go extinct in a rural area. The paper is using, what I understand correctly, is what's called a Sapphire model, which is a sort of more extended version of the SEIR model. This is a box model where you say some people are susceptible, some people are infected, some people are recovered, and you imagine probabilities of flowing between the different boxes. This is obviously a very simplistic model that it doesn't. I believe such models are not very helpful for quantitatively understanding outbreaks. They're very helpful for understanding how different factors influence an outbreak. Like they're good as a framework for understanding what's relevant to an outbreak, but they're hard for like predicting, how many people will be sick in 2 months and stuff like that. Now, in the paper, the author writes that if they imagine that people in a rural area have half to a fourth as many connections as a person in an urban area, and you just plug it into the model, then the probability of not going extinct goes down by a factor of 30. And this is a completely trivial result because the model has, I don't want to say no complexity, But it's very obvious if you lower one of the parameters, you get a lower result. It seems to be that this whole claim that it's more common in urban areas is entirely rooted on this unsourced assumption people in rural areas have fewer connections. I mean, it makes some amount of sense, but I sort of would like to see more of an argument. If we're going to get a 30 times factor here in this probability calculation, I'd like to see more than, rural people have half as many connections.
Yeah, that's a good question. And so first off, Saar and I are not in dispute over this anymore. He's already incorporated that factor in. And he's a few other things he pointed out, like it could happen in Laos or Vietnam or something like that. I did not include those in my calculation. He's correct, I probably should have. I don't know if the cuisine differs there, but for the most part, the population there is much smaller than China, so it's not really going to be a big increase overall. But if we are going to argue over this, we could do it empirically, and we could point out that the people in these regions have a seropositivity to SARS viruses. Like there was a study on Huanan villagers, which had like 3% seropositivity, but you don't have pandemics starting in Huanan. And it's hard to get the exact numbers because there aren't a lot of pandemics. We only have SARS and SARS-2. And in both of them, they started in big cities and not on these farms. So the data we do have says they always start in big cities, even though the majority of the people that are infected by seropositivity is overwhelmingly higher. So, I mean, if you just do it in ratio of antibodies, it's going to actually be a lot more than 30x. I have not actually worked with this model. It's just kind of confirming the obvious based on that data.
Okay, so a few things. If I understand correctly, SAR used a 2 times factor, and you're using a 30 times factor to represent essentially the same
No, I'm using a 2 times factor. I'm just ruling out I'm saying because it's a 30x factor, I ruled out the half of the population that's rural. And I got...
I see. Yes, that makes sense. That's a 2 times as well.
Yeah, I didn't know 30.
And then when you said we've observed the pandemic start in cities and not rural areas, when you say start, are we talking about the first observed cluster? Are we talking about where the spillover event happened? What do we mean?
First observed cluster. We never observed a spillover event for SARS. We have no idea where SARS started. There's some... Okay. They found Huanan cave bats with a virus that's 96% similar. And it took 5 years of surveillance on one cave to find that. And, Rootclaim is always saying you can't use any evidence unless you have a comparative data set everywhere. So by his reasoning, we haven't looked at any other caves for 5 years. We have no idea where the actual virus is. It could be in Hubei, it could be a different Huanan cave. Based on that connection, Xi Zhengli has a hypothesis that it spilled over in Huanan and got to Guangzhou, but there was never a Huanan case for SARS. There was never a Huanan infected civet for SARS. I came up with an argument it might have started in Hubei, but we don't have the bats there that are close enough. The difference with COVID is that we do think we have the spillover, and that's actually pretty rare for a disease.
Okay, and related to this, I'm not quite sure exactly how to phrase this question, but you're saying urban areas are more likely than rural. What I'm trying to get at is this the same phenomenon for when we've been looking at all these population maps of Wuhan? And I can't help but notice that the market is basically the most urban area of the city. I mean, is it the same phenomenon where you said, what's the chance that a randomly selected person being sick is so close to the market? Well, if the market's the urban area and you think urban is more likely than rural, is that a bias or is that not?
I don't know if you have that 30x within a city. Let me share my screen again. So a couple things. The SARS cases, I think Rootclaim said it was a farmer in Foshan. I looked that up. The very first case, he wasn't like a civet farmer or anything. It's hard to be clear on his occupation. He had animal contact, but he cooked food, which was chicken, a cat, and snake. And then these other ones, only one of them has civet exposure. So it's pretty hard to know how any of these SARS outbreaks started, only that they were in cities. As far as the density question, I think I put those up earlier. Yeah, so this is my argument there. The Huanan market is a bullseye on the market, and it's not a bullseye on. If it was like right in the middle of this area, then maybe you'd have a better case for that. I do think there's some evidence that you're like seeing more cases in those heat clusters, but there's no population density right there. And also it's a place that has something that can create a pandemic. If this was like a movie theater right there, I wouldn't be arguing for zoonosis, because it doesn't have something that you need a market or a lab, I guess. Or China says you can get frozen food, but there's not, a Mahjong room all by itself can't create a pandemic. A Mahjong room nearby raccoon dogs can. Yield.
All right, Will, do you want to do some questions, or else I can just keep going?
I have one question that might take a little bit more time. I'd like to go through the animal versus other piece that you had in your slides, because that was a fairly complex analysis, and I'm going to be honest, I need more time to absorb it, but I'd like to talk through it again and ask some specific questions.
I also am going to have questions about that, so after you go, I'll follow with related.
Totally, you can just jump in. So my understanding of this is, okay, so this is part of the steel manning the case. We're going to construct the best possible case here for what it would look like under zoonosis for the pandemic to emerge in the market. Ultimately, you conclude that even under zoonosis, the pandemic it started at the market, but for other reasons, and that's because of a variety of different factors. Is that just like the general?
Yes.
Okay, so maybe on the next slide, so the no animal vendors, and I apologize because I feel like I've seen this claim multiple times, but I don't understand. No animal vendors were infected. Do you both agree on that, or do you think that there was do we think that animal vendors were infected? Like this, it seems like Peter had data that said animal vendors were infected, or maybe can you guys just clear that one up for me?
My opinion is we can't prove that because none of them were tested. None of them, there's no antibody testing, and you only find COVID. You find 19 out of 20 you find 1 out of 20 COVID cases, and the other 19 are missed. So.
Okay. So the 5 people that those initial cases were
non-animal traders. Yeah.
Yeah. Okay.
But absence of evidence is not evidence of absence.
Okay, and then going. So you say that there's one positive raccoon dog sample, but there's only one read and negative correlation overall. Peter shares the evidence of the stall, I believe 829 or 825, where there were 5 positive frozen samples. It seems like this analysis discounts that. Why?
So I need to look back into it. I think this is about having the 20% threshold or not, but I'm not sure. I'm sure Peter would remember what's the difference, but I can check it.
Okay, but globally, it's just this you would not feel would significantly affect the analysis whether or not there was 5 or 10 or something like that.
No. In general, the important thing is that there is less SARS-2 reads associated with wildlife. Peter brought an interesting point about it possibly being due to an artifact due to different timings. We'll need to look into that. I doubt this will be enough because we're talking about an animal sitting in a cage for days with SARS-2. You will need to detect that at significant amounts, but I'll need to look. I'll wait to get all the new slides when he shares them, and I'll look into it.
Okay, that makes sense. And then final question, skip 2 slides down to the final animal estimate. Okay, so the Mahjong Hall has this very high positivity rate. It looks like it's you all differ on why the positivity rate is there or what's going on in there. What is your estimate of how much more likely it is to spread in the market, in the Mahjong hall, versus other areas? What's your probability estimate there, or what's your factor? Like, let's say a mall versus the Mahjong hall, or a mall versus Wuhan market in general.
So here the Mahjong evidence, it's not only it's existing, there's the fact that there are many cases. That's given only 30 to 40%, so it's not that big. So 2.5 to 3x. I think the market is a likely super spreader location for multiple reasons that I list, not only the Mahjong hall. The Mahjong hall I think is one of the contributing factors.
But I think, okay, totally understand. And I think I asked the question poorly, but this is building on Eric's earlier question. So imagine that sick researchers visit 100 different locations. The probability that, COVID is able to take hold in the Wuhan market in general versus the Mahjong hall versus a random mall. I think that's what I'm asking you to estimate, if that is reasonable within the framework.
All right, so the factors we're looking for are high traffic. So where is that? High traffic. So that's basically that there's a large probability that the lab worker would get there. Second is that we have permanent residents that will amplify locally, having this making this place noticeable. Otherwise, it's just people infecting all over town, so you don't notice it. And the other is having the enclosed poor ventilation in the elderly population, which make the exponent higher, right? So it will grow faster. So I think there are not many places that meet all of those. I think that seeing Xinfadi as the next outbreak is interesting. Peter made a very good point about it being traced to frozen food. That's fine, but I assume there's also deliveries of contaminated with SARS-2 going to many places in China. This is still the place that amplified it. So I think there's something about these areas that are good amplification environments.
Okay, so let me dig in. That makes a lot of sense, but let me dig in more there. So my mental conception is probably wrong of the market. My mental conception is that ventilation is reasonably high. You both agree that ventilation in these markets is very low. Is that a point of shared agreement? Like the amount of air turnover per unit time is like...
It's a closed market. This is not an open air market, yes? This is
right.
This is a closed building, yes.
I don't think I agree with that. I don't think, compare this to a bar or a restaurant. I think the ventilation is much, much worse than the person-to-person transmission. Those are much more enclosed spaces. Or the university. I don't understand how he's ruling out universities here. He says it's because they're young, so I guess you wouldn't find them, but you can still have spread, and you have residents there, you have professors, and you have students that come to class every day, so you have repeat visitors. You could say it wouldn't be found because the people are young, but we already established with the exponential math that there aren't a lot of undetected cases. There's really just these 100 cases and these 5 hospitalized. So you really still have to pick, is the first super spreader a restaurant? Is it a university? Is it the market? You only really get one of them.
Okay, to continue to focus in on this. So so we have disagreement about what the probability is of spreading given that you're in the market and what factors are there. What do you think about the train station located close by, Saar? I think of that as, again, this is intuitive, and when I was doing my own research, when I was, trying to avoid getting COVID, I looked at air handlers in trains versus air handlers in, HEPA-filtered airplanes. And it seemed like I could find no worse spot than a train to be in during COVID, just given the amount of air turnover per unit time. So do you think it is worse than a train? Do you think someone just didn't make it to a train, or is the Mahjong room possibly worse? Can you compare it in that context to my intuition?
So I think a Mahjong room is worse than a train or a bar. I think a train or a bar is worse than the market. But the important thing is that the train doesn't have the repeat visitors, the repeat population. So yeah, so the lab worker went on the train, infected 3 people next to him, but that's it. These people are now in 3 different locations across the town. They don't meet again to start the amplification in a closed environment. Okay, so they're infecting people in different places, right? And the places they're infecting are not as high exponent as the market. So you have the people returning to the same high exponent location, and that's what gives you the amplification. Because if you just infect people on the train, okay, so you infect on the train 5 people instead of 3 people that you do in the market. Cool, but still they're now gone. They're now gone, and they're now they're now infecting people in low exponent locations or 0. They're not infecting anyone, right? So you need to have this repetition, this cycle that feeds itself, this feedback loop.
Okay, that is not intuitive to me in the sense that so I understand what you're saying, where if having, if there are transmission trains, for lack of a better word, the end of transmission train occurs because people board the train and they go elsewhere and they're not in a permissible environment. That makes sense, but I think of like almost every pandemic planning scenario I can think of, or every kind of, doomsday scenario, it's somebody gets on, there's a dissemination event where you get on a train, you go somewhere and you spread out a whole bunch of different places. To back that intuition up, I guess I would say that if you get repeat visitors to the same location, in kind of a standard SIR model, more and more of those people become resistant. Your transmission probability should go down, or at least the number of hosts who are at risk goes down. So it seems to me like getting people on a train would be a much, much more likely.
I think what he's saying is you are never going to have a month-long cluster on a train, right? Like, he's not denying people can get sick on the train, but they're not going to be a cluster that extends over multiple weeks on a specific train.
But why would you? So I think the thing that I'm asking is if.
But you can
okay, so
speak
you're
for
just,
yourself.
Yeah, yeah. Now we're debating about. Is that a fair answer, Saar, to what was Eric saying?
Yeah. I think maybe I'll try to make like a more clear simulation of that, a diagram that actually shows what happens. But the idea is that you want the people returning to the same location with high infectivity. So people on the train, they're there just for 1 hour in the high infectivity environment, and then they're in the average. But in the market, you have the same people in the high infectivity environment all the time. Even if for 1 hour the train is more, this is repeat day after day for many hours. So I think that's much more important in terms of the long-term exponent.
Okay, I understand. It's the probability of producing a cluster. Okay.
Right. I think the disagreement, you're talking, Will, about the total number of cases, and Saar's talking about the chance of a specific cluster showing up.
Gotcha. Yep. Thank you.
Can I just bring up one point again that might have been missed? We keep talking about the Mahjong room as like this the cluster or the hotspot. There were no positive samples in the Mahjong room. What we have is an oversmoothed heat map that points to the Mahjong room if you look at it that way. But the actual samples were in the shops. And if you average enough of those samples together, the average centroid is the Mahjong room.
Point taken. I think I think I was trying to get to this earlier. The fact that like even the most abundant sample that has been found of a sampled animal was 130 reads at 1e⁸.
Correct.
Or 1e8. Like it doesn't seem particularly likely that you would, even if the Mahjong room was a very important place that had a bunch of spreading, that environmental samples would come up positive. Do you disagree with that?
I think if it was, the stalls where people were infected had samples in them, and the wildlife shops had samples in them that lingered until February, but the Mahjong room didn't have any at all. And I'm not saying that people didn't go in there and cough, or even that there didn't occur some human-to-human transmission. I'm just saying that people are misinterpreting a heat map to say that the samples were there when there were actually no samples there. And this is funny because we also have this heat map of the cases outside the market, which points right at the market, and they object to that because they think you can't use that kind of logic. But within the market, the heat map points to something, the Mahjong room, that they want to be the location, and they think that's a good argument. So that's pure motivated reasoning to say this style of analysis doesn't work outside the market, does work inside the market. And the center of the heat map outside, we know it's the market because the first 5 cases were there. So it's just confirming that with the heat map. If the first 5 cases were in the Mahjong room, of course that analysis would be good. But it's really just this Texas sharpshooter fallacy of, we don't want it to be the market, we want it to be the lab, so what can we put in that spot? And they tried the toilets, and then they tried the Mahjong room. And also, as I pointed out with Xinfadi market, you can confirm the bullseye on market cases with Xinfadi. That also has the centering. I can pull up more heat maps if you want me to show them on the screen to confirm what I'm saying.
Point's taken.
The actual
Understood.
bullseye is shop 629, which had the most positive samples. That's what and the ratio is low, the positivity is low. I don't know why that is. I think it might be because the animals were sampled earlier. Could be because animals, I don't know, shed less than people. I really don't know why that is. I think it's amazing that DNA can confirm, this is 12 days after the market's been closed, and maybe like more than a month after the spillovers happened, and you can still use modern DNA technology to figure out which animals were in that shop and where they were infected. And I think we kind of expect more than we should from these results, from this technology. I mean, it'd be great if you could just run a test that's, spits out the name of the animal, but that might be expecting too much.
I think I hear what you're saying. The majority of my background was in NGS used for microbial samples, so I have deep kind of knowledge of the inner workings of how that works, and that's why I'm so focused on it, is that this is that. So that's, okay. I mean, yeah.
I just want to comment that we do have multiple testimonies of Mahjong players being the early cases, so it's not just the environmental sample.
But the very first case was not, and you maybe picked 4 and 5 were maybe Mahjong players, so that's not what introduced it to the market. I provided the translated interview with Wei Guishan. She doesn't play Mahjong. So it was there before. I mean, maybe you could find an earlier Mahjong player if you looked.
Yeah, I assume she's not the first case, yes.
So what brought it into the market?
That we don't know what is the very first patient 0 in the market, but it's under lab leak it would be a person. We don't know if the person is the lab worker, it's someone he played Mahjong with, if it's family, we don't know. But the fact that so many cases are Mahjong players is something that is worth paying attention to.
Eric, did you have questions on that analysis? You're muted.
I had questions about the animal versus other slides around slide 75. If you're done asking about that, Will.
Is this one or?
Okay, so yes. So actually, can you go to the next slide, please? If I understand correctly, I'm not going to nitpick the numbers here. That's not what I'm asking. I want to understand what the numbers mean. So we have 2 hypotheses you've raised, the animal hypothesis and the other hypothesis, and several observations. We're trying to figure out if these observations are consistent with hypotheses. Are each of these probabilities, this is probability of that observation given this hypothesis, is that correct?
Yeah, so we're saying, okay, I'm telling you there's a market and an animal brought a virus there. In how many cases would you expect this to happen? In how many cases would you expect this to happen? So that is basically what these numbers say.
Okay,
it's
so
the
you're saying
conditional probability of the evidence given the hypothesis animal.
Okay, and then imagining this evidence to be uncorrelated with each other. At the end, we can multiply all these together, and we get a total probability of all of the evidence given the hypothesis. I think that's correct. I might want to double check that. And then you get a final probability of a small number. And I thought you were going to do the same thing on the other hypothesis. So we have the hypothesis for animals, and we get this conditional probability. And a hypothesis for other, and I was expecting you to go through the same lines of evidence and say probability of observing this given the hypothesis, probability of observing this given this hypothesis, and in the end multiplying and getting two different probabilities, and then you can do the ratio between them. But in the others, you chose different evidence
Yeah, I
for the
understand the
two
question.
things. So, like,
I'll explain. So,
yeah, go
so
ahead.
the idea is that all of these are expected under other, right? I don't need to estimate the probability that we won't have high infections among animal vendors, because there's no reason to expect that, right? So I don't need to reduce this hypothesis for evidence that is what is expected if there's no animal, right?
So are you saying that there's a 100% probability that sorry, I'm looking at slide 76. Yeah, is there a 100% probability that biased data is centered on the market but unbiased isn't, conditional on other?
Right. So it's
You put
to be
50%
exact, so
conditional
we're
on
not-
animal.
So what we do here is we take the important aspect of the evidence, not the specific. So having this pattern in general is something we'd say, yeah, is 100% under other. Now note that I'm also giving it 100% here in the conservative estimate, right? So of course
Sure.
it's not 100% because all kinds of things can happen. But in the essence of what this evidence, can you put mute, Peter, please? So in the essence of what this evidence is, then yeah, it's 100%.
Okay, I mean, I think it's really hard to pin down good numbers. So I imagine having a leeway of a factor of 2 here and there. I understand. So in the animal, all these things we're talking about 50%, 30%, factor 2, factor 3, and I mean I'm not ready to accept them as 100% in the other category. And so you got all these factors of 2 piling up, and individually they look innocent. You multiply them together and suddenly you have a 0.0081% probability.
Yeah,
I just
understand.
feel like there's more diligence needed to justify this comparison. Okay, but that's all I
Yeah, I
have to say.
understand what you're saying. So this is something I should have explained. So it's not the probability of this specific evidence as described, but in the essence, in the way that we have evidence that is inconsistent with an animal. What is the probability that we would have evidence inconsistent with an animal in this field? That's a probability. I'm talking about the whole possibility. And then it's, of course, 100%. So if I'm asking you, what is the probability you would get evidence inconsistent with an animal when there's no animal, then that's 100%. So I need to clarify that this is not the probability of the exact evidence, but the probability that this evidence would not be consistent with an animal.
Okay, and I guess I did have one last thing on this line, which is on slide 78. We're talking about the location superspreader. You have the numbers times 10 increase, conservatively times 4. I mean, those are not probabilities. What are they? I'm trying to
so
understand
these
what
are.
that is.
Yeah, so this is the conditional probability on the other hypothesis. So it's I should put it over there, but it kind of makes it weird. So basically, what I'm saying is that so this was about what? This was about saying that the HSM is very very rare, but we're saying no, it has a lot of features that make it a good superspreader location, especially when we consider this as a non-linear thing, right? So if a slight advantage to the market over 3 weeks would be a huge advantage. So so we're saying that what is the probability that the first superspreader location would have these features if it's an animal, right? So you can think of it as like if there's an animal infecting there, then these become less important because the animal can drive the infection. There's a source. If it's not an animal, then these things become important. Okay, so become more important. So the question is, what is the probability of having all these features? It's much more likely if there's no animal. Okay, if the reason is that a person got there, then we need these things to happen. If it's an animal, then we need them less. Okay, so that is 10x, 4x is the ratio between these conditional probabilities.
Okay,
that
so
is- that
I
is
should
the-
think of this as a 10% in the other column.
Yeah, or maybe 5% and 50%, something like that.
Right.
Yeah,
Okay,
numbers that
so,
are at.
yeah, you're
I should-
computing
I
a
should,
ratio
uh,
implicitly.
clarify these, yeah.
And I guess while I'm grilling you on what these numbers mean, I have one. My main question I'm most interested in understanding is not specific to COVID origins at all, which is your motivation behind this. You've set out from the very beginning is to sort of demonstrate a technique of thinking about and approaching problems, which I think could be called a sort of a Bayesian reasoning. I don't know if you have another label you want to use.
Probabilistic inference is the word we use.
Probabilistic inference. And you said far better than any human inference method, which is a very bold claim. And what I want to hear is I want to hear some justification, because this is either justification in general or justification specific to this application, because so as a mathematician, I'm used to, you start with some things you know to be true, and you make logical deductions. At the end, you end up with something you know to be true. And I'm ready to accept that in the real world, it's not so easy. You're never sure at any point. It becomes much harder to try to use these deductive reasoning in the real world. And I'm ready to accept, Bayes. I think we're all ready to accept Bayes' theorem for as a computational tool in cases where we know all the probabilities. It's like we know the prior probability is 10%, we know the update is 2.5, and then we can compute, given a little trouble, the correct new probability. And I will accept that it's very useful as a framework for understanding, is this evidence important? Put a number on it and then be like, oh, I guess it was only a 1.1. Maybe this evidence shouldn't be important. I shouldn't think about it too much. And maybe this evidence, there's a 1 in 12 chance. That's actually quite important. So I am very happy to accept this approach as a way of, building understanding, and forcing people to commit to probabilities and stuff like that. I'm not ready to dive into, I can compute some numbers and get a numerical answer at the end. So yeah, I'd like to imagine, suppose all of these numbers were correct. Does that mean your conclusion is correct? So I don't have one specific question. I want you to say, is your method right? Is what I want to know.
Right. Yeah. So I think the main point to bring across here is that this is about being less wrong. Okay. So it's not that this is perfect. It's that this is a methodology that has been thought about very carefully. A lot of potential weaknesses were identified and accounted for. It has stood the test of time. I think we have a track record of being in a minority opinion several times and winning. But most importantly is that people are very bad at this. Okay, so people are very bad when you give them a lot of evidence and there's a lot of uncertainty. They just mess up badly. They just get very. There's we can do some riddles and demonstrate it, but people will go way off on probabilities. That's why people don't use that, because they try a bit and then they see that it doesn't work. So this is about going through the exact weaknesses and identifying why people fail in probabilities and making sure that doesn't happen. So we make sure we don't filter evidence, and make sure we count the prior probability, and we make sure we try to get numbers that are justifiable, and if not, then we give them a wide range. So it's about not double counting and making sure you account for dependency. So there's a lot of that, and sometimes you make mistakes. But if you do this, you way better than what a human can do. That is the claim. So yeah.
Okay.
Can I add
Yeah.
add
Please
on
go
that
ahead.
as a question? So one of the things I've been struggling with too in evaluating the frameworks I think is maybe adjacent to Eric's, so not as intelligent perhaps, is how should I treat the assessments of other people? Obviously, you shared 3 Bayesian assessments that gave high probability of a lab leak. It seems like my reading of the intelligence community is that it's majority non-lab leak. It's majority zoonosis. They have access to evidence that we do not. I could construct some rather intuitive scenarios in which they have a better estimate than anything we can do here. How should I treat that kind of claim, or how should that factor into my assessment of your claims or this method?
Right. So we found very clearly that having good inference is far more important than having good evidence. Okay. So yeah, if you have a smoking gun, that happens 1 in 100 cases. Okay, cool. But usually that doesn't happen, and usually you need to be. It's much more important that you understand the evidence you have than having a bit more evidence. So first of all, I'm not sure the intelligence community does have any evidence here because, as we see, they do try to leak it, and it doesn't seem very strong. I mean, we agree with Peter that the claim of 3 sick workers is not reliable enough. We have very bad experience with evidence that's based on anonymous sources, so we don't do that. We have an experience of beating intelligence agencies, so we know that. I myself have the experience there, and I know it's at the end of the day just people writing documents. It's not, they don't have the any advanced inference system compared to people. They actually have a lot of biases. So one bias is that because they have secret information, they overweight that. So they think because others don't have it, then it's therefore stronger, but it's not. I mean, it doesn't matter that it's secret. Another thing is that in general, these groups, they suffer from groupthink, they suffer from political biases. People who choose to spend their life in intelligence agency are usually not your typical unbiased person. They would be mostly patriotic. They would be exposed to information that is mostly about we good, them bad. So in general, I have little appreciation for intelligence agency. I mean, I don't know if that's convincing.
No.
That's a lot of experience. I can go into
It's
more
very
examples,
reasonable.
but that's my experience. Something that Eric said before that was important. What was the previous question about? It was important. I forgot. Never mind. Maybe I remember. Okay.
Do you want me to go over what I said to you before?
No, I remember. It's about how to trust the numbers, and I had something to say about that, but never mind.
Should I ask Peter a question while you're thinking about that?
Oh yeah. I don't know if it will come back to me or not.
Okay.
I mean, I could talk about some of this stuff too. I have thoughts about
Yeah,
this
please go ahead.
analysis. I mean, Saar keeps saying that this is steel manning the cases, but for every one of these points, I can come up with a reason why it's about 100%, why each of these happen, or close to 100%. I mean, that would be steel manning it. I actually have some slides going over each of these. I don't want to waste all my time going over them, but if this is important, or if we extend this to the extra 30 minutes or whatever, I can do that. What I really see is like coming up with every reason possible not to believe this case and not adding similar factors for the lab. Because if I wrote out a similar analysis for the lab, I'd have to put a factor on like, okay, this employee got infected, but they didn't infect any of their coworkers. And they didn't infect any other place. And then they made it seamlessly across town. And I think you can make any of these analyses support what you want if you just add enough factors of 2. And I'll probably do this in the second debate, honestly, because I think there's a bunch of ways in which I don't think the virus looks engineered. And I can say, why would they do this? Why would they do that? Why would they put this amino acid in? And if you put enough of those in, I'm down to like 1 in a billion odds that they made this particular virus. or I'll give an exact number in the next debate. So I think this is a very fragile method of coming up with the truth because it's entirely sensitive on these, factors of 2 or other approximate numbers and the number that you add. I'd also add that he was talking about groupthink. The 3 other Bayesian analyses, I'll actually go over all of those in the third debate. These are all like drastic community. There's a huge amount of groupthink because you get status in this community for contributing to the lab leak theory, and you have to make the same kind of analysis. I'm sure there's all kinds of published papers saying that this is not the most likely outcome, even though there's a few people in this community that all say the same thing.
So I am prepared to, be skeptical of Saar's specific numbers that are going into his calculation, but I am interested, Peter, if you have a critique of the method of the overall technique. I'll be looking forward to any critiques you have to make.
Of these specific ones?
So when I asked Saar about, what did you call probabilistic inference? Is this a good technique? I'd like to hear critiques of this technique if you have them, Peter.
Oh, sure. Yeah. I think a lot of
maybe on a future date if you're low on time.
a lot of the things he's saying can apply to his own work. I was writing some of these down while he's going. Other than the Steelmanning thing, he talked about like trapped priors and how he was saying people latch onto zoonosis. I don't think that's true at all, because you can look at 2020 polls and about half of people, a little less, maybe 40%, I can look up a poll, thought it was a lab leak before we even had all this evidence, all this diffuse grant, because that's just, I was trying to demonstrate this earlier when I said, look at all these other lab leak theories. It's a normal human tendency to want to blame humans for things that go wrong in the world. And so then there's a trap prior where you latch onto that and you look for any reason why you're correct that it's the lab. I mean, there's other, like when he went over the paper and said these 4 authors are motivated, there were, I forget, 20 authors, and he picked the 4 that were the proximal origin guys, but he also could have picked Michael Warby, who suspected lab leak and really tried to disprove it. He could have picked Marianne Koopmans, who was on the WHO team, and even after the WHO investigation, she thought maybe there were outbreaks before the market because the data was inconclusive, and there was that December 8th case and stuff like that. So there's lots of other non-motivated authors. We don't even know if these first 4 are motivated. You could also talk about, like, did you read the Christian Anderson Slack messages, sir?
Not all of them, some.
Okay, so Christian at one point mentions what his priors are in there. And so he says it's a 50% odds if you count its location Wuhan and there's a furin cleavage site in it. I think those are the things. It's 50-50 lab leak. And so if that was his rational prior at that point, like it looks engineered and it's in Wuhan, but then you add the market evidence on top of that, and also the evidence he's done into, like, the backbone isn't a known backbone, and the furin cleavage site is out of frame, and there's a proline and an alanine and all these weird engineering choices, you can see how he's not lying. He just started 50-50, and then he found evidence and dropped his prior to, a much lower value.
Yield. But they did say at certain times that they don't know how this could not be engineered. So maybe it's okay that they learned how it is, but saying internally that this looks very suspicious and then ridiculing outside people who say the same is problematic. That's all I'm saying.
I don't know. I mean, I say things privately that I don't say publicly in terms of all kinds of things. I was thinking about the Nancy Pelosi, Paul Pelosi attack thing. I never that thing that came up in the news where, was he attacked by an outsider or was that his friend? Something like that. I had to look at the details. I initially suspected, who knows, maybe that's true. But I didn't go on Twitter saying, this is what I think. And there's always things that you say because they're socially acceptable, and there's also things you wonder about just because, who knows, this is a weird thing, I'm going to look at it briefly. And if you're a scientist and you know what horrible consequences are going to come if you come out immediately saying, I think it's a lab leak, maybe you're going to do the 2 weeks of investigation that they did to actually prove that it's not a lab leak before you make those explosive claims.
I understand, but still it's not about having secrets. It's about you don't ridicule other people for thinking what you thought initially. You can say, oh yeah, that is a very interesting possibility. Here's why we thought it initially, but changed our mind. But I don't think this is a very important.
And to be clear,
point.
I thought it was true. I thought it was 50-50 for a long time. And I'm not ridiculing anyone that believes this because it confused me. And, half the world is confused. But I think they're confused because there's a lot of bad actors pushing bad theories about this. There's a lot of motivated reasoning. There's a lot of outright lies, like the making up the three
It's
cases.
still not. It's still okay. I don't.
Shall we move on?
Sure.
So just a few responses. So the thing I forgot to say before is, I agree that probabilistic inference this is tough to chew. It's new. I don't expect it to be immediately convincing. In the last session, I will take all of the probabilistic stuff and turn it into something intuitive. Okay, so you don't have to rely on this alone. This is not the only claim I'll be making. I will turn it into something intuitive, but the idea is once you go through the probabilistic inference process, then you can turn it correctly into something intuitive. And I think in general, to understand what we're doing, when you do probabilistic inference, the question you're trying to answer is, which hypothesis is based on things happening that are more rare? Okay, the hypothesis that says, in order for us to be correct, the things that need to happen don't occur frequently in the past is the lower probability one. That's basically all this is about. It's about being able to deduct from the past to the present, and doing it in a way that is as much as possible free from biases and fallacies. So after we do the whole probabilistic analysis, I can then convert it into this intuitive framework, and I think it will be much clearer at that point. Just two comments about things Peter said about trapped prior. I want to be clear. Everyone has trapped prior. So the fact that the lab leak is the correct result does not mean that people that claim lab leak are doing it correctly. They're doing it for the wrong reasons. So everyone uses trapped prior. Everyone has this problem. This is not only the wrong people making mistakes. Everyone is making the mistakes, and then sometimes people happen to be on the right side. So I'm not trying to claim that lab leak proponents are free from biases, mistake, or anything like that. The rest is not important.
Okay. I've asked a lot of questions. I still have no shortage of questions. Will, if you want to put in a few, also, I can either start skipping questions for Peter, or we can extend the time.
I'm open to.
Let's get to the point where it's 0 and then decide.
I would like to ask one for Saar here on the kind of general. I think one of the difficulties is there are competing methods of coming to these conclusions. Like, I think I can articulate all of Peter's claims in a probabilistic inference fashion. Like, I don't think there's any, I think you all are not disagreeing on that being a reasonable method. The question is what gets included and what gets excluded there in terms of the calculations. I'd like to have, one of the perhaps easy biases is to believe the zoonosis, because I can, as you said before, I can come to a kind of an intuitive picture of how that works. It passes from a bat to something. I can see the person taking the train going to the Wuhan market. I kind of focused some of my questions on your proposal about trying to build for myself a similarly intuitive and simple picture of how it might work in your model. And I think you kind of wanted to say, like, we don't take a position on how it might have happened, whether or not it's, left via, or whether there was an environmental exposure that led to a lab leak, et cetera, et cetera. So that's the backdrop of this question. What I want to ask is, what has to be true about the activities that were going on at WIV to make the production of the SARS-CoV-2 probable? So you cited a statistic that said it was, Dr. Barrick says it's $6,000 to create a novel virus. The diffuse proposal doesn't get funded. How many viruses did they have to make? What did they have to do such that one like this leaked? Give me a little bit more about how you think that occurred.
Okay, so first of all, I think that's a great question to ask Yuri. Next session,
Okay.
he will be able to do it much better from memory. But basically, we already know they collected and sequenced many viruses in nature, so they already have that in the database. So going ahead and trying to find the ones with the best ACE2 binding, that's not a big deal. That's something that they can do. They already have what they need. Yeah, it costs money, of course, but that's what they do. That's that's their job. And basically, what I have here is these are basically the activities that will result in SARS-2.
So I guess that makes sense, but I'm thinking kind of I'm trying to constrain, or I'm trying to build a picture of how surprised should I be? Like, okay, so let's say that WIV had $10 billion and they were doing Golden Gate assemblies and mixing every spike and every ACE2 together. I would not be particularly surprised that they came up with a very infectious virus. It doesn't bear on the question of whether it leaked, but I wouldn't be surprised about that. But given that their grant wasn't funded, and we agree that it's fairly expensive to do this, and given my knowledge of how researchers operate, where, you can get some stuff done without a grant, but you can't get a large genetic engineering project, give me a scenario or give me a constraint, or reject the idea that I can constrain the analysis in that way. It just seems like they would have to spend a bunch of money to produce this thing. They didn't get their grant funded. It seems like that would go against the probability that this was actually, mix and match Golden Gate assembly, something happens.
Okay, right. So first of all, the important thing is that we know there are bat viruses in nature with the RBD similar to SARS-2. So we know those exist with this level of ACE2 match. So it's all about whether in the 180 viruses they sequence, whether it's there or not. Okay, once they're there, then how costly it is to find it. You can start with simulations. That could be cheap, and then you go in vitro. So I don't know exactly how much this will cost. We can look into it. It's very probable that they took the same proposal and gave it to the Chinese government and got it funded. I don't know, but we do know that people are there doing their job, and we do know that is the kind of stuff they find interesting in 2019. So yeah, I expect this could happen. I mean, not 100%, but 1.2% per year, sure.
Okay, so that I understand what you're saying. One more to sharpen it, I guess, even more. It seems like that explanation relies on one of those, that the SARS-CoV-2 being a direct chimera, or it seems like the probability is highest under that explanation that it's direct chimera. So if that's not true, but would you agree that the more intermediate steps are required, the more there has to be a funding source, the more there should be some sort of paper trail, something like that? Like, what I'm trying to get you to do is to constrain how much of a project this had to be.
Right. So currently, so we don't know if it's a chimera or not. Probably not. But basically what we know is if you take Banal52, a virus that exists in nature, and put an FCS in it, you're pretty much at SARS-2. That is not a lot of steps. So this is a virus that exists in nature. You just plug in the FCS as it is now, and you're done. You don't need to do anything else. This becomes infectious. The lab worker gets it under BSL-2, and that's it. So there's not that much genetic engineering going on here.
Interesting. Okay.
I think this is for Yuri much better. Keep it for the next session.
Totally, yeah, absolutely. Go ahead, Aaron.
Okay, the last few of my starred questions for Saar. Now, in Peter's presentation, he not just gave a critique of the lab leak theory, but he also critiqued the credibility of some of the people putting forth the lab leak theory, so on the one hand, you don't want to be in the company of Steve Bannon and Rudy Giuliani. This is not good company. But on the other hand, what does it matter what other people are saying? What matters is what you're saying, right? I want to know, do you feel like you need to respond to Peter's criticism of other lab leak theories, of other proponents of lab leak theory, do you feel like you're in bad company? Do you feel like your word stands strength on its own?
Right. So of course, we propose a specific hypothesis which is the Steelman of the lab leak position. We compare it to the Steelman of the zoonosis position. We don't care what anyone else says. We have been in many cases supporting people that are very fringe. It's just the way the world is. So truth is hard. It's not always with the people you like. Sometimes the people you don't like got it. And again, it's all by chance. I mean, everyone is using bad inference. So it's not that there's no good reason why this happens one time that way and another time the other way. We don't care what people say. We don't use that. Maybe if I'll have a few minutes left that they need to just talk freely, then I can go through my my responses to Peter's deck, and you can see like we have this happening all the time. Human inference ignored. Like whenever the claim is about some people say something, that's fine. We don't. It's not something that affects the probability of the hypothesis.
Okay, wait.
People will say weird things always.
Let me jump in there. So I saw these in the human inference ignored, but what struck me is, and I think this gets to one of Peter's questions, is you rely on the 1977, the Russian flu, the H1N1 Russian flu, as a evidence or as your prior for flus. But that seems totally human inference based. It seems like like I found competing theories about what caused that. You say lab leak, but that seems totally human inference. Like how do you not have to be accountable for that?
Good point. I agree. Yeah. So eventually, at the bottom level, you need to use human inference. The whole idea of this process is to break down the very intractable questions into smaller pieces, where human inference is generally not that bad. So if whether we should have counted 1977 maybe as a half case instead of one case, that could have been a mistake, and we should fix that. I'm sure there are other mistakes. I'm sure there are places where we were too conservative and we need to bring the numbers up. The idea is that you give this leeway both sides and make sure that you leave room for all the mistakes, and eventually you get a number that is robust. Here it's important that we have, to this point, the prior plus the WIV activity and Wuhan. We have two different methods that reach very similar numbers, and I think, I can maybe I'll stand corrected, but I think they don't look very biased or motivated. I'm sure Peter will be able to show where I'm wrong here, but I think these are two methods that are generally balanced, Steelman both sides. But yeah, let's see. Maybe I'm wrong.
Okay.
I can respond right now if
go ahead.
If you want. I need to share my slides out. Is that up? Yeah. So I thought this, my mic's on, yeah. So the calculation on the left isn't bad. It does miss two human coronavirus that happened since SARS-2, but that's not huge. I think the odds of Wuhan might be a little higher than 1.5. The lab leak rate, I looked this up in a few papers. They said about 1 in 500 per lab per year. Maybe you can make that higher if you think the WIV is unsafe, but I don't think you're going to make 100 times higher, but maybe, you need to have proof for that claim. I think the odds they have a secret starting virus are much lower than that. I don't actually know where this 180 unpublished viruses come from. We need to talk about this. Diffuse as written couldn't make SARS-2. I found a video clip of Yuri Deigin even agreeing with this. He says you have to go beyond what's written in the diffuse grant to create SARS-CoV-2 because it's done in an abnormal backbone, And there's also these other things like the foreign cleavage site doesn't look natural. It's hard to find a way to culture it successfully. And so, yeah, the 15% seems way too high, and the 50% seems way too high. And there's a bunch of other factors you can add in this. And also pandemic starting in Wuhan, that's like 30% versus 40%, but that's not a big deal. But I think there's a lot we can argue about here that you're missing. Yield.
Okay, I'll give a quick response. So 1 in 500 is the general, studies about generally lab leak, those that are found. Here we're talking about the situation where we already went through the steps, and we have SARS-2, and we have someone working on it on BSL-2. I can easily say that why this is close to 100% infection, but I think 15% is pretty conservative. So this is a very different case compared to the general background of people working with viruses. These are people working with an extremely contagious virus in very bad conditions, specifically not protecting their respiratory tract. And about the FCS, backbone, all of that, that's discussed next session. These are evidence that are not in the model at that point. So maybe they increase, maybe they decrease. This is just evidence up to that point.
That was actually going to be exactly my question, which is, I understand not protecting the respiratory tract is a bad idea, but what lab procedures would be aerosolizing or getting the virus into the air? I imagine most people are working with the virus in like bulk liquid or not in. I don't see how the virus gets into the air to breathe in. I want to understand how that happens.
So one thing they describe is working with is infecting animals with the viruses they produce. So that's a very easy way to get it. I don't know enough about other procedures. Yuri could probably give you a good sense.
I think that's unfortunately easy. I think it is.
Okay.
Having stuck myself with a couple of needles and with a couple of pathogens.
Okay, I guess related to that, I recall in your analysis, you gave the factor of 4x for the BSL-2, which I sort of quibbled with a little bit at the time. I think I can better explain my quibble now, which is, are you saying that other viruses are getting studied in greater safety? Is that what that's representing? Because your prior probability that a release, that a pandemic is caused by a lab leak already takes into account the general safety that people works with viruses in labs. So if they worked with SARS-CoV-2 with biosafety level 2, but they work with everything else also with biosafety level 2, then shouldn't that already be accounted?
So it's a good point that the priors describe situations which were probably less safe than normal. So that's a good point. But I think here we're talking about something that is far more extreme. So this is something that should probably be at least BSL-3, and it wasn't BSL-2. So remember that SARS has escaped from BSL-3 and BSL-4, right? So these are tricky viruses. I think giving this factor makes sense. To try to maybe substantiate it harder.
So the 4x represents increased probability of lab leak of this specific virus compared to a typical virus.
Yeah, not the typical virus compared to virus that leaked in the past. So which those were also safety breaches. Right? So it's not compared. Of course, SARS-2 and BSL-2 is probably 1,000 times worse than just what's happening now in any normal virology lab. That's crazy. But compared to past lab leaks, okay, so 4x and 2x conservatively.
Okay, I'm down to pretty much my small questions. So, Will, if you have anything else you want to do before we run out their time?
I have one kind of high-level question, which I pose to both of you, which is, and you can refuse to answer, but I think I want to know what the most important 3 claims you think you need to win are, so I can understand where I'm thinking about the evidence as presented versus where you all are thinking about the evidence as presented. And perhaps this is something that occurs at the next debate, or we need to extend time, but I'd like to understand what you think the 3 most important claims are for each of you that you need to win. If you have questions that need to take precedence, Eric, that's fine.
I'd like more time for that, or say that for next debate.
Okay.
I can give some ideas. So I think we are not that far, I think, not that far in priors. So if it's 20% or 8%, that's fine. We're not that far about Wuhan as a location. It's also fine. I think the important points would be our claim that the market, with all the evidence around it, makes it an unlikely patient zero location. That's something that's probably important to determine. We will probably be a big issue about whether the furring cleavage site insertion is natural or not. That is something that is, I think it's like very, very strong evidence. We're putting it, it's something pretty conservative, but it could easily be 1000x evidence. What else could be important?
And I'm most interested in just this debate, Saar.
For this one, yeah, the market, I think, is the only thing that's really a big issue here. Yeah, priors probably we can improve a bit, but I don't think we're that far apart, and the location pretty similar. So yeah, the market is the big thing. Whether our claim that the evidence of the market is so problematic that it's better to just assume that it's a coincidence than an animal.
Will,
Great.
do you want to finish any other questions you have, and then I'll just use the rest of the time? Okay.
All you.
So I got 2 minutes, Peter.
Can I use my 2 for some closing statements, or are we? I think you should decide now if we're extending, because otherwise I'm going to want to use my time differently.
I'd be happy to talk about like a 15-minute each extension. How do you feel, sir?
It's 3 a.m. already. Let's do
Okay.
10 minutes.
We'll have other times to talk.
Yeah,
so
let's do 10 minutes.
I mean, okay. Sorry, 10 minutes total or 10 minutes each?
Each.
Okay. And maybe we won't use it, hopefully. I'll try to go quickly. Peter, I got a few sort of related questions. I'll just give it all.
My time away. No more. Ask me your question.
Okay. So I want to talk about the significance of the most recent common ancestor. It's a moving target, literally, because as time goes on and different lines die out, the most recent common ancestor gets bumped up further and further. So there's a few points where I'm unclear. Are we talking about the most recent common ancestor of, the cases that existed in Wuhan in early January? You're talking about the most recent common ancestor of cases today. Are we talking about, the first spillover? Is there a reason why? It seems to be that you're arguing that the market is both the first major cluster and where the spillover itself happened, although you don't seem 100% sure, but you think that's the most likely explanation. So is there a reason we should be getting a cluster of cases where the spillover happened? And I'm going to round this out with one last thing, which is you're arguing the case, the evidence shows it's impossible that a September, say, origin happened because of the long time lag. And I'm not completely convinced that the evidence shows that decisively, because it seems unlikely to me that the virus was circulating at a very low level without going extinct and without creating any great outbreaks for 2 months, passing from person to person at a low level. But a pandemic is a really rare event. So I don't think it's, to me, completely out of the question that the process to get a pandemic involved some rare things that go along. Now, obviously, if the virus emerged in humans in September and was as contagious as it was in January, then yeah, I'm accepting that's impossible. But it's possible that the virus that first hit humans was much less contagious. It passed at a low level, miraculously didn't go extinct, but picked up some mutations that made it more contagious among humans. And this is independently of whether we're talking about zoonotic or lab leak, that when it first passed into humans, it might have rapidly become more contagious. So that's sort of a bunch of related things, and I just want to know what thoughts you have about the relationship between when the spillover happened and when the first cluster showed up.
For some reason I'm having trouble sharing my screen again, so I do have some more slides for this, but we've seen the last slides already. Most recent common ancestor is a moving target. Like if you did it now, I think you would find like the Omicron first case would be the most common ancestor, because everything else is, other than cryptic lineages, everything else is gone. But it's quite stable until Omicron emerges. If you try it at any point between the market outbreak and then, you get the same result. And could you have cryptic transmission in Wuhan for 2 months? I can work out the exact odds for you based on these models. It's extremely unlikely, but it depends on your model. Can you have cryptic transmission elsewhere in China and then it suddenly hops to Wuhan on a train? Maybe. I haven't thought too heavily on that. We don't see like this rapid evolution once it jumps over into people, and we'll talk about that at the second debate. It seems, but also it doesn't rapidly evolve like when it spills over into mink or deer. So that's probably just like this generalist property of the virus that both allowed it to spill over, but also allowed it to not mutate that much. It did have some early adaptations, actually, though. It had the D614G mutation that enhanced the furin cleavage site. That happened very quickly and had a great improvement in the efficacy of the virus. So that argues both against prolonged culturing of the virus in a lab and prolonged transmission of the virus in humans.
May I propose a scenario to you? Thanks, Will. Imagine that the virus that spilled over in September did not look like any of the early samples that we have today, and it spread with a very low but positive growth. And maybe it spread in multiple places in China, maybe it spread in multiple places in Wuhan, and then it happened to reach the market around the same time. It developed a very positive mutation that blew it up. I think you gave a doubling time of like every 3.5 days, or a small number of days.
So that scenario is inconsistent with a lab leak, because if you're saying it has this furin cleavage site and this ACE2 binding, you're definitely, that virus is ready to go. So if the lab did do that, it's not going to circulate for 2 months.
I just want to understand if this is consistent with any explanation.
Could it have, is there some natural explanation where it circulates widely and then it suddenly gains the furin cleavage site in the market? I'd have to think about that some more, but off the top of my head, I don't see why that's impossible. I don't see why that's more likely, but...
Yeah, so the ancestral virus would still exist. It'd just be rapidly outcompeted by the descendant virus, and then all evidence the ancestral virus is lost, and we're left here with, I guess it...
So something important about Picard's simulations about the polytomies, the two basal polytomies, that's the pattern, which is where you have this root, so like your root's lineage B, and you have a bunch of mutations coming off of that root. And so you can tell what the most recent common ancestor is because it's the base of that polytomy. And so if it was actually, like lineage A and lineage B can both be the base. There's arguments for both. But like there's this other argument for ProCove2, which we're going to talk about in the second debate, where that's one mutation even further to the bat side, and I'm going to say that's a reversion, and they're going to say that's ancestral, and like that really can't easily be the most recent common ancestor, because if that was it, then the base of the polytomy would be Prokov 2, not lineage B.
Okay, let's see if I have any questions for Saar that I've. Something really quick. Saar, do you disagree, or are you happy with the distinction that Peter made about a super spreading event versus a cluster? He said that this market was not a super spreading event; it was a cluster. Is this important to you? Do you have an opinion?
No, I think I agree. If I recall correctly, super spreading means that there's one person doing all the work. I don't think was that the distinction, Peter? Whether it's one person or just the location?
I don't know if that factors into our models. It's definitely a slow burn through the market, not a super spreader, in my opinion. I guess we're in agreement on that. And
so this is about how rapid it was. I have no idea. I didn't look into that.
It's not like one person from the lab goes into the Mahjong room and then suddenly it's 60 cases. It's 1 week there's 3 and then the next week there's 6.
Okay,
like that.
Yeah.
And it's weird if that slow burn is happening at the market, but not anywhere else in Wuhan. That's why I think it's not a super spreader location. I think it's really weird that's the only place that slow burn's happening.
It sounds like there's not a disagreement anyhow. I just wanted to make sure you had a chance to say something in case you disagreed with what Peter was saying.
I'm not sure if this maybe I'm missing something. So it doesn't seem like the market has happened like with one person infecting 60 in one day. That doesn't seem to be the case. But definitely the market conditions created a higher exponent in the market. That's our hypothesis.
I don't see any disagreement between you there. Okay, another thing for Saar. I think at one point around slide 49, you were critiquing certain papers that are against the lab leak theory. Ahead of time, I don't understand the significance of these papers. So I have a sort of a dual question here. One is, do you think that the accuracy of these papers is important to Peter's argument? And the other is, do you have critiques with the papers beyond critiques with the authors of the papers? Like, do you see specific problems with the papers, or you're just concerned about the author's method because the
You're talking about 39? 39? Let me share it.
I wrote down slide 49. Let me just look at it.
This?
Slide, I guess 47.
Oh, okay. So just to clarify, this is about ferrine cleavage sites being a focus of virology. I don't have any critique of these papers. This is just to show a pattern.
Slide 47?
This one?
Yes. So you mentioned these papers, and then you go on to give messages that the authors of these papers gave.
Yes.
And you're concerned about the accuracy of these papers. I'm wondering if you have concerns within the substance.
Yes. So the substance is all of these things. This is lots of things that go against the paper.
So that's this. Okay, I understand.
Yeah, and this is just about saying you need to be very careful when there's like heavy data analysis by people who want a certain conclusion to be correct, whether intentionally or not. That's all.
Okay. On slide 51, you said before January 18th, they needed a connection to the market for a diagnosis. And I want to know, when did that start? Because obviously, at some point, they didn't know about the market. So there must have been a starting period when... And also...
I think this whole thing started December 31st. I don't remember.
And
maybe Peter seems to be much better memory than me.
Who is beholden to these recommendations? Is this like a specific hospital is doing this? Is this all of China?
So I think what happens is they send like a kind of message to all all hospitals that this is something that's going on. Pay attention. This was from the CDC, I think. The CDC issued this message. I need to check it. Can you clarify your question? I'll answer it offline.
I guess the trouble is that I'm not clear. So both of you at many different points talk about, the cases that we know about, cases we know about at the time, cases we know about retrospectively, and the sampling biases. And I think it's actually quite hard for me to get a handle on where there are sampling biases and what the source of the sampling biases are. And I just in general, I want to have a better idea of exactly, and this is related to the fact that many of the times both of you showed maps of like case counts, and I'm never sure like what times. Are we talking about cases in December? Are we talking about cases through the whole Wuhan outbreak?
Okay, so I'll tell you my impression. So we tried to dig into early cases, and it's a mess. I mean, there's so much information, so many contradictions. There's this 200 paper report just about early cases, each hospital, what happened. This is not something we think is reasonable to analyze with confidence, and I don't think it's important because initially we thought the market was an important coincidence. At this point, we're fine with saying that the market is the only big cluster at the time, so it doesn't really matter. So so we can just accept that, and we don't care about biases or no.
Okay, and then I guess I was going to ask a few slides later. You talked about someone who's diagnosed only because of a relative. I guess you're saying this is not important. I wanted to ask. Chen? Yes.
So Chen. So this is a bit different. So there's a difference between saying that the market is the only cluster. That's one thing, and it's very different from saying that the market is everything. Okay. So we're definitely saying that there are a significant number of cases outside the market. Okay, that's definitely something we're saying. We're just saying that they didn't manage to create a big cluster anywhere. Okay, so definitely Chen is important because Chen shows you have someone pretty early, which cannot be connected to the market, and we know the bias. We know why he was noticed because he has a friend in the central hospital, and the central hospitals are the ones that have the better sequencing abilities, right? So we think this case illustrates that there are a few dozen cases at least around Wuhan that no one knows about.
Just a quick question. Do you know when the first case outside of Wuhan was? Sorry.
I don't remember now.
It's Beijing on December 17th. So if it can get to Beijing in 6 days, and that guy was actually market linked, is how we know about it. So we're sure it's exported from the market. But if it can get, across China in 6 days or 7 days, I think it can make it across Wuhan in 1 day less than that.
Right.
Yeah, but I don't think it's weird that there's someone who was to the market and then went to Beijing. That happens. The question is, we have someone who says, I'm not going anywhere. I'm just living in a suburb.
Yeah.
And can't point to anything that's related to the market. And he's 16 or 8 December case.
My question
about it's been spreading for a few weeks. So you don't think there will be any cases in town that aren't market linked?
I'm not saying there won't be. I'm saying that the fact that we have this case and we know in that case specifically, there's a good reason why we know about it because they go to Central Hospital. That's a coincidence that you need to point out because why don't we have 3 other cases in suburban clinics, right?
But it seems like you're going out of your way to ignore the 174 cases, but you're focusing on the one case as disproven.
So this is all of December, right? By December 16, I don't think we have 174, right?
Correct. So he's maybe the first non-market linked, and there's always going to be a first non-market linked.
Yeah, sure. Of course, it's going to be a first non-market link, but it happens to be the one where he has a good reason to go to the central hospital, which others would not. So that is a significant.
If it wasn't the market, it would be 100 to 1 cases that aren't linked. This proves that it's really That's the thing. If there's 100 cases and there's 5 hospitalized at the market, then it's the market, because that's the hospitalization rate. And if it's not 100 cases, if it's 1,000 cases, then you should expect a lot more non-market linked at that point in time.
Right, but if it's 200,
Sure, yeah, you can make up some
okay.
number that fits in that
No,
window.
it's not about making up a number.
The-
That's what
the
the evidence
only-
reads, right?
The only question I had is, do you know when he was diagnosed?
Yeah, I can look it up.
Because
So
I'm assuming he wasn't diagnosed on December 16th because nobody
Yeah, I
knew
can
what
look it up.
COVID was on December 16th.
yeah, so
So he
it
must have been
somewhere. I can look
diagnosed
it up.
retrospectively. It wasn't a very
I'll
important
look it
question.
up. Continue in the meantime.
Okay, let's see, and maybe,
I would, Eric, if you're okay, I would argue that we move towards giving closing unless you have some other pressing bits.
okay, let me just,
We're pushing up on 6:15 now, or I mean 6 hours 15 minutes.
okay.
Chen
yeah,
was
there's one
sequenced on December 26th. I don't know if that's considered diagnosis, but he was sequenced, so I assumed.
That's good enough. One last small question on slide 85. You talk about Saar talks about breach of containment is more likely during the move of the Wuhan CDC, and I just want to know if you have any information about whether bat coronaviruses would have been involved in this move, because it sounded like from what Peter said that there wouldn't, in which case the move should not be significant in that respect. If you have any information about like materials being moved, not just people, that would be relevant to the breach of containment.
Is this to me? The question is to me?
Yes.
I don't know. We were just hypothesizing that such a move would be disruptive in that sense. We don't have specific information, and again, this is like really like the last line of defense. I don't know if we're
Okay.
going to get there, but I can try and see if there's. I don't know how we can find it's not the kind
Yeah,
of information you can find online, but
I just
Think
want to make
about
sure that everything potentially contentious gets challenged. So,
okay.
okay. And then I guess I have a few small questions I want to run through for Peter, if you're okay, before we switch to
Yeah.
coding.
Sorry, one thing I just wanted to answer of your previous question, the question about bias. My claim is that the search was unbiased before December 29th in the early hospitals, because no one has publicly announced that there's the connection. And I'm saying that the search is biased after December 29th for some period, and you can tell that the percent market cases go up, actually, market linked. And then on January 18th, then they remove the market linked, and the percentage goes back down again. So I'm saying it's more or less unbiased after that. And I also pointed out that 127 of the 174 were diagnosed after that was removed. You can still, hypothesize some other form of bias, and it's hard because it has to fit the whole part where most of the cases they find are unlinked, and many of them, or some of them at least, are very far away. So I don't really know what form of bias would find that. Also, Warby's paper considered every form of bias as one of their arguments. They tried just sequentially removing the cases closest to the market to see how many you could remove before the association was lost. And you could remove, I have a slide for it, you can remove like half to 2/3, depending on how you're doing the math here, before you lose the association, and it just matches equally well with the population density center. So even if you could imagine there's some other form of bias, it still has to be extremely strong before it's relevant.
Okay. So, Peter, quick question. Maybe this is more appropriate for next session. Do you have any thoughts on whether the Wuhan Institute of Virology might have been inadequately handling materials that were contagious? Because this was important to Saar's argument that the situation is there for a lab leak. I want to know, do you agree?
I don't know. I think it's probably exaggerated, but I don't think my argument really relies on that. I think most of my argument is the many lines of reasoning that show that the most recent common ancestor is at the market, and the geographical association with the market, the early cases with the market, multiple lines of reasoning linking the outbreak to the market, not the lab, and then you just have this very rare coincidence of how it got there. And then on the lab side in week 2, I'm going to point out things like all their work was done in different backbones. Like normally they plug a spike into a WIV1 backbone because they're concerned with SARS viruses and they want to know what happens if you tweak SARS, things like that. And the foreign cleavage site doesn't look natural. So I don't think my argument is really going to rely too heavily on I know what their exact procedures were.
Okay. And I think...
Also, they do this work in pseudoviruses frequently, which can't infect people. That's something that's neglected in this debate. The whole gain of function is rising. You also need to account for the fact that we have advanced experimental techniques like pseudoviruses where you can do more experimentation safer.
Okay. We'll probably hear more about that next time, I guess.
Yeah.
And then my last question
Okay, we'll do it next time, yeah.
Is you claimed you attributed some significance to the fact that SARS-1 and SARS-2 spilled over in the same month. And I want to know if there a reason why spillover events should be seasonal as opposed to just the virus.
I wrote 1 in 12 in my probabilities, and that's definitely too high. I figured we were going to negotiate that down. I'm happy to say 1 in 6. I think Saar even said 1 in 4. And I wasn't trying to pull one over on you guys. I was ready to argue that down. I could make the same argument November versus December. It is important to realize that COVID is not a particularly seasonal virus. It can spread in summer. I just caught it in summer. And it could leak at any month of the year. So we're looking for every piece of evidence we can. That's a small one. I don't think that factor of 4. I think there's a problem here in that both Saar and I think our arguments are very strong, and it would take this overwhelming evidence to overturn them. And I don't know if we're going to end this debate actually not believing that at the rate we're going, but I'm optimistic that we can make some progress in seeing, where we both agree and disagree.
There was a slight distinction I'm making. I'm not asking about whether COVID is seasonal. I'm asking whether spillover events should be seasonal. I'm not aware of any particular reason they should be.
Well, so on the human side, it would remember how we boosted cities because of the density,
Yes.
and deprioritized rural areas. So you also are going to have higher transmission rates in winter, and that's going to be an extra factor on that 30x. And that might be why, even though it could spill over multiple times, but it's going to stick better as a pandemic in winter.
I see.
But that might be why.
I'm a little reluctant because I feel like biology has not definitively come to understand exactly why flu virus and so on are seasonal. And to go from flu virus is seasonal to COVID pandemic and spillover is seasonal, I'd feel better about it if I understood where we started.
Yeah, we don't.
Okay.
So we only have a few data points, and actually HKU1 seems to also be a winter in China thing, but that one's really hard to contact trace, so we don't know it as well as SARS or SARS-2. So if you want to drop the factor of 4, I'm not going to be angry. If you want to drop the factor of 10,000 because SARS says the market's equally likely to be the animals or the Mahjong room, then I've done a bad job making my case today, or we need to talk about that more.
I want to ask there just because I thought there was a basis of the claim. I thought it was a bat immune system claim for spillover likelihood or transmissibility likelihood. Not that it's important, but did I miss
I guess
it?
if more bats are sick, then more spillover can happen. Is that?
That's what I thought.
It's not crazy.
I'm trying to understand the coincidence, and I don't know if it's important. I did find papers explaining that there is a sharp spike in bat virus seasonality in September, October, and Huanan, I believe, was the cave they were looking at.
Okay. Not super important.
But I don't know how fast it gets from bats to intermediate to human. So I don't, there's a lot of unknowns here, and we don't know that for SARS. We don't know that for COVID. We don't know that. We don't even know the intermediate for HKU1.
Okay. Well, thank you for your patience with all my questions. That was most of them.
Thank you.
So if you want to give any closing arguments.
Me? Sure. I have 2 minutes. Let me see if I can share my screen again. Can you yield the screen, Saar, or whoever's
Sorry.
sharing? So I think this is one of the key things that we were talking about, and I don't think Saar is steel manning here. So the first point, 15%, I worked that out in the Zhao Zhao paper as 7 of 17 shops are this market. So yeah, I could say it could be another market. There's 4 of them. This one is actually the one with the most shops, but you could call that, lower, 41%. And I don't see why TCM hospitals are relevant. Restaurants maybe, depends. He said, no infected vendor when really it's 1 of 7 for the SARS outbreak. So, and this is even harder to contact trace. No animals were tested, so I don't know what the relevance of not finding a animal is, or no relevant animals. Best explanation, he's ignored the 2 spillovers.
1 minute.
Every excuse of bias he's come up with has fallen apart so far as I can tell, so I don't understand why he's still using that. Lineage A, and the Mahjong board, is not relevant as far as I've demonstrated. So steel manning these, and not giving these 100% necessarily, but just making up as many examples why you don't like it, I don't think is a convincing argument against it. What do I have, 30 seconds? Yeah, I think he's making up a lot of numbers here. And I went over this one. This would be good to keep working on though in the next debate because these are really important. And I would close with a quote from Mark Twain. There are 3 kinds of lies: lies, damned lies, and statistics.
Will, you're muted, but I'm assuming you're handing control over to Saar.
Yes, sorry. 8 minutes and 32 seconds.
And sorry, just to be clear, I think if there's leftover time when you're done with conclusion, you'll just have it next time.
Sorry, what do you suggest?
I was saying if there's leftover time from your concluding marks, then we'll just adjust it next time.
I'll just go quickly over my. I will go quickly over my comments on clock, over my comments on Peter's presentation. So just go over the main problems. So there was some issues about Wuhan being claimed to be an unlikely. That lab leak people claim Wuhan is an unlikely. We're not saying that. We're just saying it's one of. It's an equal probability we give it. That's not important. It's important to remember all the differences from SARS-1 that had multiple spillovers. It had more genetic diversity at the start. We'll discuss next time. It's clear how it got to Guangdong. We had the positive sample, so important to look also at the differences and not only similarities. So there are several claims that making Hubei especially likely. I mean, in order to determine that, we need to see an objective comparison of all the provinces under the parameters and see that Hubei comes up. I mean, of course, in retrospect, you can find anything you want that is specific to Hubei. What else is interesting here? Saying that WIV is far from the market, it's not too important because we don't expect the lab to be where the infection starts, but actually where the lab worker frequents. We discussed that we need to add other countries. So here we accepted the density and added the 2x, so that's a correction we made. So here there's a claim that Hubei is not banning events. This is for the time being, and then later we see that the whole of China stopped banning wildlife after SARS-2. So probably, these things. A lot of these things that try to single out Hubei seem to me to be cherry-picked in retrospect. I'm willing to be proven otherwise. Just need to see a process whereby you compare all the provinces and see what happens. Okay. Okay, this is an important one. So this is the calculation that eventually Peter does that goes to the billions. So want to see so we did this very similar calculation that produces the 1.2% per year, and the main differences are having a suitable virus. So given what we know about Banal 52 and the missing unpublished viruses, I think it's likely that they would have one. It's a problem they created because that's what diffuse says. So once they decide to do diffuse, then it's they're likely to reach a virus like that. Probability of escape given BSL2 we discussed, very likely, and spreading in Wuhan. Make it across town to the market without spreading elsewhere. We don't agree that is the case. And about the lab successfully hitting all the evidence, we will discuss the behavioral evidence in the last session. But definitely, we're not talking billions here. This is something that is pretty much likely. Important thing is that Peter said that we're claiming that it happened in a BSL2 lab, but we're saying BSL4 is WFV having BSL4 makes Wuhan more unique. So this is a very important distinction. BSL4 is a proxy for the extent of virology research. The fact that they have BSL4 in Wuhan means that that's an important hub for virology. The fact that they specifically did SARS-2 and BSL2, that makes the case worse, not better. It doesn't bring up all the BSL2s across China to be relevant. The BSL4 is only an indication. We're looking for an indication of how unique is Wuhan in that sense. And in that sense, the BSL4 is an indication. We're using much stronger evidence for that in our analysis, but just important to understand what BSL4 is about. So the work of WFV is much more significant than any of these. So any case where, ridiculing people who make weird lab leak theories, that's fine with us. People are weird. That's not a problem. Okay. Okay. Yeah, so the example of using cholera for centering the source of the epidemic, this is very different because cholera isn't airborne. It doesn't have much human-to-human transmission. Many of these people were actually infected directly by the water pump. Yeah, and it's also before we had advanced transportation in cities. So this whole case and equating it to the centering of cases in Huanan is very shaky. So these are many options. This is the option. None of these is the option we reached at. Conditions of the market make it a good superspreading location. There's possibly one of several, but there's no dispute to biases. So the last sentence, we're willing to concede it, even though we think it's correct. It's not important. Okay. We talked about that. So this is like, okay, there's a lot of work to do in early cases. If it comes down to that, we'll probably need to have a whole session just on that. It's very complicated. We've talked about that. Here, this is, we showed it as a table. Here you see it as a map that actually the district of the market is lower than its surrounding in terms of seropositivity. We talked about the importance of taking into account the other map of all-cause mortality, not only pneumonia. Yeah, so the whole issue about the moat and valley, how's it called? Yeah, we're not doing that. Our CDC claim is very specific. Yeah, all the things about dates we don't find very important. It could eventually come up to be important, but we'll if we get there. This was interesting, the fact that there wasn't any event that would could be an alternative to why Wuhan was important. So we didn't know that. So that's strengthening the case that WAV is responsible compared to something else. Yeah, I think I'm running out of time there. So yeah, that's about it. See you in 2 weeks.
In my last 6 seconds, all inference is human inference.
All right, wow, thank you very much. This was quite the marathon session today.
Thanks, that was fun. Thanks for the patience. You guys are great.
That was awesome, guys. It's really cool to see two people who put in so much work. And,
Definitely.
And thank you, Saar, for making this contest. This is, like, there's no place to, like, challenge these beliefs, you know, except on Twitter, and Twitter conversation is terrible, and it's fun to actually talk and, like, figure out what's right and wrong, and I don't know.
Thank you for challenging. It's really, you're the first one to ever go the distance.
The only guy dumb enough.
Huh? We'll see. Okay.
Yeah.
Okay, quickly.